Good afternoon, everyone, and thank you for joining us for today's workshop and module. Today is Module 4 of our Probabilistic Genotyping workshop series. Before we do get started, I would like to go over a few housekeeping items with those of you that have come into the room. Those of you that have just joined us, the first thing I would like to make mention of is our chat pod, which is located on the left-hand side of your screen marked chat. This chat pod will be the best way to communicate with us if you have any questions regarding today's material. All questions pertaining to the material will be retained until the Q&A session that will be held either after each presenter speaks if we have the time. If not, we will hold all questions until the very end of all of the presentations today, and we will have a panel-style discussion Q&A at the very end. So please make sure at any point of today's module to go ahead and type in your questions there in the chat, and we will pull them out and hold them for you, and then we will get them answered during our Q&A. This chat pod is also the best way to communicate with us should you experience any technical issues. If you do have any technical issues, please make sure to let us know there in the chat, and we will be sure to get you taken care of. All of your audio for today's event is going to be streaming through your computer speakers. So if you can hear the sound of my voice, I'm going to ask that you take a few moments to utilize the chat pod on the left-hand side of your screen and let me know that you are not experiencing any technical issues thus far. We are utilizing our new platform as we have for the entire month of May. So just a few things to point out. The best user experience is going to be on Mozilla Firefox or Google Chrome. So please make sure you are utilizing one of those platforms. Also, there should be a play button on your live feed window there in the middle of your screen. It's either in the middle of it or on some browsers it might be on one of the bottom left or right corners. Make sure you do hit your play button in order for your live feed to begin. And then if for any reason you lose slides or audio throughout the presentation, it might be due to a connection issue. You might need to refresh your page and then simply hit the start button again. We do like to find out a little bit more about our audience before we do get started, so if you could take a few moments just to type in your location there in the chat pod, that'll give us some great feedback about our audience and just the kind of reach that we are able to accomplish with today's module. Again, this is Module 4 of our series. We're very excited to host our presenters and subject matter experts today, and we will get started here shortly. Thank you for joining us. Good afternoon, everyone, and thank you for joining us for today's event. Today is Module 4 of our Probabilistic Genotyping Workshop Series. So we will get started here in just a couple of minutes, but before we do, I would like to go over a few housekeeping items with those of you that have joined us in the room. For those of you that we do have with us, the first thing I would like to make mention of is our chat pod, which is located on the left-hand side of your screen marked chat. This chat pod will be the best way to communicate with us if you have any questions regarding today's material. Please feel free at any point of any discussion or topic today to go ahead and type in the questions that you might have for our subject matter experts, and we will house those until the end of the presentation, or if we have a large number of questions, we will hold all of the questions until the very end, and then have a panel discussion Q&A style discussion. So please make sure that at any point of today's webinar or today's workshop to go ahead and type in your questions there in the chat pod, and we will hold them for you to get answered. This chat pod is also the best way to communicate with us should you experience any technical issues. If you do have any technical issues, please make sure to let us know there in the chat pod, and either myself or one of our customer service representatives will make sure to get you taken care of. All of your audio for today's event is going to be streaming through your computer speakers. So if you can hear the sound of my voice, I'd like for you to let me know in the chat pod that you aren't having any technical issues thus far. That will also verify that the chat pod is working properly for you as well. Please feel free at any point if you do lose audio or have any audio issues to go ahead and type them there in the chat pod, and we will be sure to get you taken care of. Some things with our new platform, we do want to make sure that you have the best user experience possible. So please make sure that you are using Mozilla Firefox or Google Chrome as your web browser if you are able to. These will give you the best user experience for today's event. Also, if for any point of today's event you lose your presentation slides or your audio, simply refresh your web browser and then hit the play button in the live feed again, and that should refresh your connection. We are going to get started here in just a few moments. But before we do, and while we're switching everyone's layouts, if you could take a few moments to type in your location there in the chat pod, that'll give us some great feedback about our audience and who is joining us today. Again, we are going to go ahead and get started. I'm going to switch some layouts around, and then we will bring on our first speakers here shortly. Thank you for joining us today. Good afternoon, everyone, and thank you for joining us for Module 4 of our Probabilistic Genotyping Workshop Series. My name is Josh Vickers, and I'm with RTI International's Forensic Technology Center of Excellence. Today we have 3 presenters and subject matter experts, Dr. Tamara Moretti, Dr. Sarah Noel, and Dr. Duncan Taylor. We'll do our quick disclaimer. The opinions and findings and conclusions or recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. Now, I would like to introduce the brains behind the entire workshop series, Dr. Tamara Moretti with the FBI Laboratory. She has developed this series as a forum for experts in the field of statistical genetics and probabilistic genotyping to share their knowledge and experience as the forensic DNA testing community transitions to ProbGen. She will be moderating today's module, and I would like to go ahead and hand things over to Dr. Tamara Moretti.
Thank you so much, Josh. While I try to switch over here to share my screen with you, I want to welcome you all to today's session. Thanks so much for attending. If this is your first webinar with us, or if you are returning. We've done a number of these in the past few weeks, starting back on May 1st, where we heard some of the background issues, some history, and some of the things that we deal with in the forensic field with interpreting complex mixtures and other types of evidentiary DNA typing results. And then we moved on to learn about statistical aspects of probabilistic genotyping. And last week, we saw some of the developers of 3 software programs present to us about those programs. And Michael Coble gave us a comparison of the results from some of those ProbGen programs. So today we want to move into validation of these probabilistic genotyping systems. And then we're going to take a break next week and then continue back on June 12th. So I want to begin, first of all, by thanking Jerry Laporte from NIJ and his staff for supporting this webinar, and also Josh Vickers and his colleagues there at RTI for helping to make this whole process happen for us. I'm thrilled today that I'm going to be joined by two fabulous speakers that I've invited to speak today because they just give really good presentations about their research and their activities in their laboratories relating to probabilistic genotyping. I'm going to talk for the first couple hours, and then we're going to take a little break. And then after me, Dr. Sarah Noel is going to present. Dr. Noel received her PhD in biological sciences from the Université de Montréal in 2009. She's been working as a DNA analyst at the Laboratoire de Sciences Judiciaires et de Médecine Légale in Montreal since 2011, where she's been responsible for hundreds of cases. She has conducted a large research project regarding DNA transfer in the washing machine and the persistence of semen stains laundered multiple times. And that's something that we've been discussing here about transfer and persistence of DNA evidence. More recently, she acquainted herself with probabilistic genotyping and has been working on the validation of star mix for Casework. She's also participated in the creation and distribution of extensive internal training material for our star mix implementation. So thank you so much, Sarah, for joining us today, and we're looking forward to your talk. I'm also thrilled that we're joined by Dr. Duncan Taylor. He is a principal scientist of forensic statistics at Forensic Science, South Australia. He holds a PhD in molecular biology from Flinders University, and he has worked in this forensic field for over a decade, supervising criminal casework and hundreds of cases and testifying in courts around Australia. Dr. Taylor is one of the developers of StarMix, which we've seen last week as one of the interpretation softwares that's being used by forensic DNA testing laboratories around the world. He's also an associate professor of biology at Flinders University, where he supervises honors, masters, and PhD students. And there he is working on pioneering new methods of evidence evaluation to assist the judicial justice system. So thank you as well, Duncan. I'm glad you're here to lecture to us today. So I'm going to move into my presentation, which is going to be a long one. It's got basically two parts. Or three, about the validation of probabilistic genotyping systems. I'm going to be going through the SWGDAM guidelines for validating ProbGen systems, and while I have captured a lot of the information just by cut and pasting into my PowerPoint, I would refer you to the actual document if you want to, for more information, or for, I'm just hoping today to share the information and provide some examples from our validation studies, and then as well to talk about some specific topics such as sensitivity and specificity and precision of ProbGen systems, especially for LRs close to 1. Now at the FBI, we implemented probabilistic genotyping in December of 2015 with an STR kit that we already had in usage, and we were reporting random match probabilities and CPIs, combined probability of inclusions, with that particular kit when we implemented our first ProbGen system. And then a year later, with the expansion of the CODIS core loci, we had validated a new AMP kit and then revalidated, at that time, an update to our ProbGen software. And I've listed here just a number of things that are just my personal opinion of what our experience has been working with the program that we use. Now, we saw last week there were three softwares that were presented. And all of those developers obviously have the adequate background and experience as developers and forensic science and statisticians. Some of the software programs that are out there have seen plenty of casework usage for a number of years. They have extensive publications. They're not black boxes. We may be challenged, I think, at times to understand some of the things that the software is doing, but with the publications and the training opportunities, hopefully we're able to arrive at an understanding of how the systems are operating, and certainly to the extent that we can explain them in court. To that end, the next couple webinars are going to be focused on communicating LRs and court-related issues. We've experienced exceptional training and support with this continuous system that we used. It's based on widely used and accepted mathematical and statistical and population genetic principles. We like the fact that the biological modeling is perfectly consistent with our historical approaches, that the parameters that are used in the software are based on our in-house data, that we can define our populations for allele frequencies, and that the performance features and the IT requirements for the software fit right in with what we had in place in the laboratory. And then note another thing about our discussion today, that validation of ProbGen systems is very specific to the kit that is being used, the detection platform, the CE instrument, and the conditions such as PCR cycle number and injection conditions. So when I talk about validation of software, this big change that we made in 2015, our IT folks at the FBI laboratory saw it a little differently. When I say the word validation, I've gotten a sense that the people in the IT arena view it a little differently. But really what, I don't know, looking up information about IT validation, it seems to be somewhat in line with what we are doing. It's demonstrating that the product fulfills its intended use when it's placed into its intended environment. What we're talking about, though, is going to be very specific to our forensic application, and I'm going to be speaking about the SWGDAM guidelines for validation of a ProbGen system. And there are other guidelines or publications out there, or maybe information from the software developers that I would encourage you to read as well, because the more information you have, the better. So when you see the little SWGDAM logo or seal up at the top, that indicates that I'm taking information right off of the SWGDAM guidelines, which you can find at swgdam.org under the publications section. So what these guidelines say, first of all, is something that Mike Coble a few weeks ago underscored for us, is that it's really necessary that you strive to have an appropriate foundation knowledge and the calculation and interpretation of likelihood ratios before going into the usage of or the validation of probabilistic genotyping, because the results from your validation studies are going to be likelihood ratios as well as other information. So we like to understand that aspect of the data so that we are prepared to evaluate the validation results. It's up to the laboratories to determine which validation studies are relevant to the methodology and, in the context of its application, how to demonstrate the reliability of the system and its potential limitations. The laboratory also must determine the number of samples required to satisfy each guideline and may determine that a study is not necessary. For example, we at the FBI laboratory have two different protocols. One allows us to use different parameters for missing persons cases. So that particular application obviously is not one where we would need to evaluate mixtures when we're dealing with bone specimens. So moving on into some of the details of validating a ProbGen system. First of all, SWGDAM says the laboratory shall validate a system prior to usage. Now, when the SWGDAM guidelines use the word shall, that's something that you have to do under the guidelines. And shalls are used where it's also something that is in the quality assurance standards for forensic DNA testing laboratories. If it's not in the standards, we typically say that something should be done. You shall document all the validation studies in accordance with the QAS, and you should have access or document things about the software, how it performs, its operations and activities, what the methods of analysis and statistical formulas are, what is the input data, what are the operations that are performed by the user, what's the system workflow, and what are the outputs. So it's necessary that we understand all of this about our system because it helps us define exactly what it is we need to test in order to adequately validate the system. This guidelines also speak about system control. You need to have adequate computers to run the software or servers and verify that it's properly installed, the configures are correct, and some levels of system control, such as documenting unique version numbers, only allowing authorized users to access the software, audit trails to track changes to the system data, or verification of the system settings and your casework each time a run is performed. Also, system users only are allowed to perform authorized actions. Now, before going any further into guidelines, I want to take a little break from reading all the guideline slides to show you what I typically present at the very end, because it's sort of at the end of the process, and that is forensic case studies. So what the guidelines say about case studies is that laboratories with existing interpretation procedures should compare the results of ProbGen and manual interpretation of the same data. Now, we recognize that these are completely different systems and not directly comparable. They're based on different assumptions, thresholds, and formulas. But what we're looking for is your manual interpretation process to generate results that are generally consistent from what happens with probabilistic genotyping. These are data from our case study for our first implementation of our ProbGen software, where we're looking at CPI versus the LR with 19 case profiles, and there are 4 references or persons of interest that were used. And I know it's complicated, so I'm going to walk through how this is set up. Here we have above the line the log LRs, which basically is a representation of how many zeros are on these statistics. And above the line, those would be the inclusionary ones that support HP, and below the line, the exclusionary ones that support HD. And by the way, we at the FBI laboratory use the terms H1 and H2, and I might use those interchangeably with the kind of the historical vernacular for the propositions, but those are what we're referring to with those terms. And down at the bottom is just captured what the software returns as an LR of 0. And so that line going across horizontally basically represents a million, where if you were using a verbal scale, you might describe that as very strong support above and below the lower one. So we read the results in columns like this. So this is for an evidence item that's designated Q19, and this is actually bank robbery evidence where we have a lot of clothing and a bottle. And these are 3- and 4-person mixtures. And what we are looking at is down here, this is actually 2 outright exclusions of LR of zeros that are overlaid. And this is a second POI, and this is, a third and a 4th POI, and the codes are up here. This is a major contributor that's returning a really high LR through the ProbGen system, and green means it was reported as a major contributor, and that would have been with a random match probability. And this symbol represents a CPI. So in that instance, this person could not be excluded as a possible contributor to this mixture. And all the gray bars basically are that same scenario, just consistent with the previous typing results and nothing terribly remarkable other than that. Over here, what I've shown in green, though, is something interesting. Again, 2 LR zeros at the bottom, another contributor that was previously excluded that showed support for HD. And then this represents something that was inconclusive in the manual interpretation. And so this would have been interpreted using an analytical threshold, and I'm sure a number of those loci were not suitable for usage, but we were able to outright exclude manually 3 of the subjects and this with this particular item. But this one reported as no comparisons. It was inconclusive. See how high the LR is for that particular item when you're able to not filter out data using a stochastic threshold and use all of the data in the probabilistic genotyping approach. And then there were 2 other evidence items that followed that same basic trend where inconclusive results. We were now able to use the data and it supported the prosecution hypothesis. The next category is shown here where we have a number of exclusions previously, and that particular one was returned an LR of 0. I'm sorry, an LR of 1. So that was uninformative, whereas previously we had reported that as an exclusion. And then for this particular one, we had that example, which was reported as an exclusion before, where alleles that were identified by the probabilistic genotyping approach were disqualified in the original reported one because they fell under the stutter filters. So using the better biological modeling of stutter, as we now have with our probabilistic software, the result returned an LR of 400, and this was a trace contributor comprising 5% of the mixture. And then the last grouping that I want to discuss has some interesting results here. These 2 individuals could not be excluded before, whereas they're providing support for HD at this time. And then there's other points there. So let's look at those in further detail here. What we have is the item number, the references, the number of contributors to the mixtures, and then the ones that you see in the shades of blue or gray are all supporting H1, and you can see the CPI statistic and what the HPD LR is. And if you were to look at the red-colored rows, these are the 4 that were circled there, and this would be this number supporting H2, or the 1 over this number would be the HPD LR. The CPI in each of these instances was 1 in 1, and that is the statistic that would be returned in PopStats along with an indication of the percent of the population that would be included. So this 1 in 1 for the first evidence item on the last screen included 75% of the population, whereas the others below included 98% of the population. So I know this statistic, labs have different policies about whether they would actually report data that is this common, if you will, but our policy has always been that it is a conclusive result. They were conclusive profiles, and it's just that a lot of people would have been included, the predominance of the population, and we are able to obtain support for HD with our probabilistic system. So back to the guidelines, the laboratory should determine whether the results produced by the ProbGen software are intuitive and consistent with expectations based on the non-probabilistic methods analysis. Based on the non-probabilistic mixture analysis methods. And I think what we just saw, we saw some differences at the last, but bear in mind that we're using better systems at this approach this time that are able to use more data. So I don't think those are inconsistencies of the kind that would that would be problematic for the validation of the software. It's an improvement in the software. Here we have a demonstration of our prior kit, and then that we had tested with our ProbGen software, and then our expanded locus kit. And as Mike Coble, if you saw his presentation, he showed how to read these particular graphs when you have this line of equivalency. If data points fell on this line, basically you have the same result here and same result here. That would mean there are no differences. And when the results fall up here, this is basically demonstrating exactly what we would expect, that we get higher LRs with the usage of an expanded locus, quite a bit higher. And then there's 3 data points over there on the left shown in yellow. Those were 3 mixtures that returned HD support for a known contributor with the 13 locus kit, but they correctly provided inclusionary support when we used the expanded locus kit. So more information is better in this instance. Going back to the guidelines, now let's step back to the introduction and background. So depending on the performance characteristics of the software, prerequisite studies may be required. So I'm going to go through in the next few slides some of the prerequisite studies that we performed at the FBI laboratory. And the first has to do with the analytical threshold. I think we're all familiar with the concept of the analytical threshold and how it is established. But what we discovered early with our usage of our probabilistic genotyping system was that usage of the global filter in the GeneMapper IDX software, which we had typically set at 2%, this could cause problems with probabilistic genotyping, our particular program. And the reason was that the global filter was removing labels from peaks that the program was expecting to be there in its modeling process. So we no longer use the global filter. And because of that, we tend to see a number of artifactual peaks that are very low on the RFU scale. We then took a data set of 106 samples and tested, or basically counted the number of edits that were required if we use various analytical thresholds. And based on this information, we decided that we have to go up all the way up to 150 for our analytical threshold. And as I mentioned before, if we do that and determine for, say, a missing persons case that we have a really degraded or low-level sample, and it's a single source, bones, teeth, hair, and tissue, that we can drop the analytical threshold. But if that's the case, we then need to factor in drop-in, which I will come to that in a moment. Now on to the topic of sensitivity and allele dropout. The manufacturers have mentioned to me that they have been asked to develop more sensitive kits. And as forensic scientists, sure, we want to be able to get DNA evidence off of evidence when it's in fact there. But we have to be careful what we ask for, because these more sensitive kits, as Peter Gill talked about in our first session, there's the issue of background DNA that we could be detecting, or DNA that has nothing to do with the crime that's present at very low levels. So our kits have become more sensitive, and that raises the question of, should we be making sure that we're not over-amplifying, if you will, such that we have a good degree of allele drop-in? I note that the instruments that we have been moving toward, the 3500, has a much greater sensitivity and a much greater dynamic range. Here's our data with green showing the manufacturer's recommended cycle number and the various loci. The sensitivity of detection there, we're getting results that are around 10,000 RFU. And especially at that level, we had been experiencing, in our validation study, we've been seeing significant spectral issues that were complicating the interpretation of mixtures in particular. It's difficult when you have so much bleed through or pull up, especially if something falls in a bin, for determining what could and couldn't be an allelic peak. It can be impossible to do sometimes. So we didn't want that high degree of sensitivity, and also tested 28 cycles and 27 cycles of amplification. I showed this slide on our first webinar, I believe. Just wanted to come back to it too. These are data from our allele dropout study, where we're looking at the low end of the results at the various cycle numbers. And typically at this level in the past, we were seeing a fair amount of allele dropout, which is shown for a single dropped allele as a yellow cell, and for the loss of both alleles at heterozygous locus shown in red. Going down the scale, we are now looking at data from a mere 10 cells. And look how many of those profiles, and by the way, we're looking at a single PCR for a single sample would be this row going across the top and so forth. Look how many samples are generating full typing results. And even if we were to pick through this, we could certainly see even down at about the equivalent of 5 cells, full typing results can even be obtained at this level, and so forth, where we start to see more dropout. We felt that this level was plenty sensitive, but we had to assess this in the context as well as allele drop-in. Before I move on to that, though, I wanted to share this information. This is a result from a 29-cycle amplification of 62 picograms. So it's exhibiting your classic stochastic effects where we've got some allele dropout, we've got locus dropout, we have intralocus peak imbalance. And I want to direct your attention over here to what's going on at SE33. And so this might be something that we would feel comfortable calling a 1319. Well, what this sample truly is a 1419, and this is what I refer to as stochastic stutter. What's happened is that the stutter peak was generated fairly early in the PCR and took off and was amplified, and for stochastic reason, the actual allele was not amplified nearly as much or to a detectable level. So the 1419 at this high cycle number, this low template sample would have been called incorrectly. So how to generate all the data? On a few of my slides, I've put some information which would cue you into the number of samples we might have tested at the FBI. This is what I call the foundation study because the data can be used for so many things, including the information that we use for ProbGen software to set the parameters. We would amplify 25 donors in a dilution series and in duplicate up top. We used to do it all in duplicate, but we encountered an instance right after setting our stochastic threshold in our validation. Right after we set it based on duplicates of everything, we had a exception to the stochastic threshold. So we now amplify this in quadruplicate to ensure that we've generated plenty of data down on this low end for setting appropriate dropout parameters. With respect to allele drop-in, we've discussed that in one of the past webinars. It's a non-reproducible, unexplained low-level peak observed in a profile, and we can measure the rate at which it occurs in extraction blanks and samples. Those are the best ones, and also look at our negative controls. Here's the data that we used, and we did see slightly higher drop-in rates with a higher cycle number. Here you see the rates. We tested the 3 different cycles and then did a little more work on the one that we were interested in using and included our robotic processing procedures and generating these data. But because we have set our analytical threshold at 150, we don't currently model drop-in with our settings for probabilistic genotyping unless we have a missing persons bone type of case where we need to drop the AT down to 50, and we would then model drop in that way. And there is a cap. This is actually the highest. This was, I think, was a drop-in peak that might have had some spectral issues associated with it. We re-injected it a number of times, and it's really sort of around 100 or so. So we set a maximum expectation for drop-in peaks at 150 RFU, which basically tells you as well that we would not expect to see drop-in the settings that we use for analysis. And then the last prerequisite for setting up a ProbGen system that I'm going to speak about relating to validation is this issue of saturation. You know, with CE, the amount of DNA that you have amplified in a sample is reflected in the amount of fluorescence in your electropherogram. Too much DNA, though, can saturate the detector. And beyond this value for probabilistic genotyping, the model is no longer optimal. So how do you determine what that saturation point is? The following is a clever approach that was shared with me by Joe Bright. And so this is how it is calculated. I'm basically showing here a number of peaks that are at 10%. And here we have 1,000 stutter to a source peak that's at 10,000, 2,000 stutter to a source peak that's at 20,000, and so forth. And then at a certain point, though, we will reach, with increasing amounts of template, the limit of detection on the instrument. So this peak is technically, in my hypothetical example here, it's technically 4,000 RFU, but the instrument can only detect it at a certain level. It's saturated the camera and thinks it's around, say, 30,000 RFU. And so if you were to do the math here, 4,000 for the stutter peak relative to a 30,000 RFU source peak is a stutter percentage of 13%. So it appears elevated in that sense, but it's actually not. We're just not able to detect that source peak accurately. So what does this have to do with saturation? Data can be used in the following way. If you take the observed stutter and the stutter ratio, you can then calculate what the expected height is of a source peak. And then if you were to plot this in one of those graphs, I'll show you next with the diagonal line. So we're basically going to be plotting observed on one scale with expected on the other scale. So this is going to fall right on the line, as do these. At a certain point, we'll see the 30,000 plotted relative to the 40,000, and you'll see this occur. So on this line of equivalency, that tells you we are on scale. We haven't saturated the camera, and at a certain point on the RFU scale, here it happens to be in the FBI data, 30,000 RFU, the point at which we saturated the instrument. So this is just another one of those prerequisites for setting up your ProbGen system, or it could be depending on what software system that you are using. And last, stutter. So a lot of work goes into establishing what your stutter expectations are. Here's an example where we have some reverse stutter and forward stutter. And I am going to cheat a little bit and draw a peak in there artificially like that. Now play along with me, because if this were heterozygote, I would expect probably the heights would be maybe about half what we have here. But for demonstration purposes, that new peak that I've added into the profile is going to have its own forward and reverse stutter, because this locus does have forward and reverse stutter. So we might expect that to occur. And there is some discussion about whether peaks of that nature are, in fact, additive. I believe that they are. In my experience, there might be some variation just due to run-to-run differences or stochastic effects down at this level affecting the peak height, but I believe they are additive. So we might see a peak of that height in this position. So that is something that perhaps in a manual approach to evaluate at certain loci in this situation, this additive stutter position, now that we have forward stutter occurring as well, might cause a peak to exceed what we would have historically used as a stutter threshold. But our probabilistic software, if it's able to model both reverse and forward stutter, would potentially be able to take that into account. And then, of course, there are other stutter positions that we are familiar with. The biological modeling now is done in a much more sophisticated way, so that if we, for example, had what I've shown there as an X as a result in a profile, whereas we might have manually filtered that and, with a guideline of 11% here at the D3 S1358 locus, ProbGen-wise, it would know that that's got some allelic component to it. But so what the software, a software program might use an approach at something of this nature, where we have basically a line of fit for the data here, and what falls on the line is shown in red here, which you could actually calculate with the equation for the line that's shown down here at the bottom. But you'll notice around this line, clearly there's some variance. Okay, this allele, or this stutter, does not occur at the same exact point. So the ProbGen software might use something like these variance curves that are shown on the right in its modeling of a phenomenon such as stutter or the actual peak heights, the allelic peaks. So what this shows us is that we have a small proportion of data where there is low variability, and a small proportion of stutter, this is the stutter variance, a small proportion has high variability, but most have this sort of mild or moderate level of variability. And we also, this is the data for the forward stutter and so forth. And on to peak height ratio. Another way to calculate that or think of that is as heterozygote balance, just another term. But for our ProbGen software, it's calculated a little differently. This is the exact same data, just calculated a little different. The low over the high RFU allele, which generates percentages or numbers less than 100, or the high molecular weight always over the low molecular weight, which would give you values above and below the line here. And no matter how you look at it, we see that with increasing peak height, the variance is going to decrease. And then if we were to look at that same data and apply there in gray our conventional thresholds for heterozygote balance, you see how much tighter the result is now, which is, again, refining our ability to use the exact same data in analyzing profiles. So whether we're talking about stutter variance, allele variance, the variance at different loci and their efficiency of amplifying, which equates to balance or imbalance within a locus or among loci, there are a number of things that will impact these results. Sample quality effects, pipetting of the extract or the PCR, how much template you have, lots and lot differences, what's going on in thermocycling, the biochemistry or CE prep, the edge effect. There's an edge effect of where the sample is on the plate as you present it for capillary electrophoresis and so on. All of these things are things that translate into what your profiles look like. So the question is, for the different software programs, as we learn about them and understand what they're doing, what does the program need to know about your data and what to expect from your data? Now, if I'm using 28 cycles and you're using 29 for the same kit, even though we have the same CE instrument or same model, our data is going to look a little different. But perhaps if two labs are using the same approach, then their data is going to look similar. And that's one of the features that allows us in multi-laboratory systems to work off of some of the same information. A good data set, then, I would suggest, a good data set be used to establish these parameters, such as that foundation study that I showed before. Because if you, for example, were to do a small experiment, you might have an instance where there wasn't a lot of variance, or if you just performed it on one of your CEs, if you have multiple CEs. And if your variance is set too low, it's basically requiring the data, in order for it to fit, it has to behave very similarly. And in the process of MCMC, it's going to take more time to find those accepts and extend the analysis time of probabilistic genotyping. Permits very little difference from the settings, and that could result into false exclusions. If you were to generate a small data set, you might then just randomly have a data set that has a high level of variance. That could result in a slight lowering of the likelihood ratio and lead to false inclusion. So the message here is just to generate enough data in your validation and understand what your system is doing. Now I'm going to move on to developmental validation, and I'm not going to show any data, obviously, but I'm just going to go through some of the requirements of those who we look for in terms of developmental validation. Defined, it's the acquisition of test data to verify the functionality of the system, the accuracy of statistical calculations and other results, the appropriateness of analytical and statistical parameters, and the determination of limitations, typically done by a manufacturer or developer, and it should also demonstrate any known or potential limitations of the system. The underlying scientific principles of the method and characteristics of the software should be published in peer-reviewed scientific journal. That's what we look for. I would have loved for this to have been a must, but we certainly can't dictate what the journals will and won't accept. So we would like this information to be peer-reviewed if possible and made available to us. And the sorts of things that you can see there on the screen are the things that we would like to know about how the software is operating. Developmental validation should address sensitivity, specificity, and precision. And I'm not going to describe those at this time because I'm going to go through them in great detail as we move on to internal validation. It also requires case type samples. If the software is designed to assess controls, those should be evaluated. In terms of accuracy, we would look to see the accuracy of calculations as well as any allele designation functions the software might have. These studies should include comparison to the results produced manually or to an alternate program. And if the software uses raw data files from a genetic analyzer as input data, the peak sizing, calling, and allele designation functions should be compared to the results of another software program in order to assess the accuracy of those functions of the system. Now on to internal validation. This is the accumulation of test data within the laboratory to demonstrate that the established parameters, the things that we've talked about up to this point, software settings, formulas, algorithms, and functions perform as expected. And in accordance with the QAS, internal validation data may be shared by all locations in a multi-laboratory system. But there would, of course, be performance verification requirements for those off-site laboratories or those other laboratories. Now, depending on the features and capabilities of the software, some DNA typing results may or may not be determined to be suitable for such analysis. That's part of the process, something that the lab has to look for. To identify these features, such as the minimum quality requirements or the number of contributors that a laboratory will be allowed to process in Casework through ProbGen, to identify data features that render a profile appropriate or inappropriate for ProbGen, the laboratory should test data across a range of characteristics that are representative of what is typically encountered by the testing lab. Okay, the data should be generated in-house with whatever amplification kit or instrumentation for detection and analysis software is used. There are mixture data that are out there available for testing and so forth, but for the process of validating a ProbGen software, you need to use data that you generate in-house, and I think you've seen why based on some of the earlier information we've seen. Some studies may be conducted using artificially created or altered input files. I can think of, for example, we had very few instances of, actually no, instances of drop-in in our sample containing profiles. And so we took the input files and just manually added artificial drop-in peaks in order to assess the software's capability to address drop-in. Okay. And in terms of the limitations, data should be selected to test the system's capabilities and identify its limitations. In particular, complex mixtures, low-level contributors should be evaluated thoroughly. And that helps us to define the limitations of the software, as well as things that we may deem not suitable for computer. So some things that may be among the more challenging ones are similar contributor ratios, 1-to-1 to something. We may have, if you have, say, a major contributor with and a minor contributor that have alleles that differ in size by one base, like a tall 9.3 etho1 and a short 10 allele that kind of blends into the shoulder of that peak. That's a good test. And also minor contributors and the height range of stutter peaks. Now 3 different topics we're going to go through that are part of internal validation. The first one is precision. So precision studies should evaluate the variation in likelihood ratios calculated from repeated analyses of the same input data. And you should do this with various sample types, not just a single source, but different types of samples. Some ProbGen approaches may not produce the same LR from repeat analyses, and we've heard about this so far with respect to the MCMC. So where applicable, these studies should therefore demonstrate the range of LR values that can be expected from multiple analyses of the same data and are the basis for establishing acceptable variation in the LRs. So the MCMC process, Dr. Buckleton, I think, was the one who introduced us to that, where the arrows were moving along and exploring the probability space. And this is a sampling procedure, and therefore the weights will vary slightly between each different run of the software. Now, the MCMC process, though, where it does introduce this variability, it's a small source of variability compared to some of the other ones that I mentioned a moment ago, such as those from the PCR process or CE process. And, in the software we use, this variability, as well as that of the allele frequency database, is taken into account by a highest posterior density method. This is basically a type of a confidence interval and is the LR that we report. And I'll show you some examples of that in the next few slides. So the extent of this variability was investigated by interpreting 2-, 3-, and 4-person mixtures where there was ambiguity in the genotype combinations of the minor contributors. And in these instances, we took the same data and repeated the ProbGen analysis 10 times. So this is a 2-person mixture, and we're zooming in on the scale so you can see it a little bit better. So you can see here that, if I could find my cursor, here we go, that we've got this variability in the LR, and this is the HPD LR, the lower bound, if you will, which is the one that we would report. So the idea here with this lower bound is that any one of these should be lower than 99% of the LRs that would be generated. And here we have The precision demonstrated for 3P, three-person mixture, zoomed in, as you can see the amount of variability, and 4P. So we find from this part of the validation that the LRs in our software program are very reproducible and that the lower 99% bound of the HPD is appropriate. One of the more interesting questions, though, and I think we'll circle back to this topic when we talk about the SWGDAM guidelines or recommendations for reporting LRs in a couple weeks, is precision when the LRs are close to 1. So James Smiley at our laboratory at the FBI ran a whole bunch of mixtures in StarMix where we had basically data. I'm going to show you data going closer and closer to 1 as we go through the slides. And where I've outlined them in red, these are just our validation samples where one of the contributors was degraded. So you can see here, we're in the range of 200 to 400, okay, and I have scaled everything to the same scale, so they're all comparable here. And then we're going to go down the scale. This is for HPD LRs from the 60 to 100 range. So you can see how much variability there is there. Now we're down to 10 to 40, and we still have everything on the same side of 1. And then let's see, now we're down to LRs that are between 4 and 11, and the HPD LRs are still supporting HP. We're getting really close to the value there. But as you would expect, you can eventually take a lower bound that is going to go to 1 or even cross over. Well, there'll be some instances where you have data that is less than 1. So this would support HD, whereas the others would either be greater than 1 or at 1. I actually can skip through the next couple slides because it's all summarized here. So we have from those precision studies, these are all of the mixtures that had LRs of 10 and less and where the HPDs fell. And I'm going to transition it, though, because we truncate these values for Casework. So you can see that we would have some values, even though the data are, the LRs are very reproducible, the precision is really high, we might have some instances where something that is greater than 1, the HPD LR is at 1, or there were a couple instances where the result would have been less than 1. SWGDAM's position on this is that the lower bound is the appropriate measure, and if the lower bound is an appropriate representation of the result here, there's no reason that we should consider a lower bound inappropriate just because of where the value falls relative to the scale. So this truly is an appropriate representation of the data as well. So in terms of the profile features that are recommended under the SWGDAM internal validation guidelines, specimens with known contributors, as well as case type specimens that may include unknown contributors. The value of the known contributors, of course, is that we know the ground truth. If your lab does anything like changes cycle number or injection conditions, you should assess or include in your test data variable DNA typing conditions, alleles that have a variety of peak heights, including off-scale peaks, obviously single source and mixed specimens of various contributor ratios and various template amounts and various numbers of contributors. Now, your laboratory might decide that we've heard about software that can handle up to 10-person mixtures. Your laboratory may decide with that software that you do or don't want to go up to that number of contributors. So whatever, you may want to test them all, maybe not, but you need to have certainly tested anything that you would want to be able to report on or analyze in Casework. If the number of contributors is input by the analyst, you should try using both correct and incorrect values. The data should have examples of allele sharing among contributors and partial profiles that have allele and locus dropout, DNA degradation, perhaps inhibition. You need to test allele drop-in if that's something that the software models as well. Forward and reverse stutter at any number of positions that the software handles. Intra and interlocus peak height variation, and additional challenge testing you might think of, such as what happens if you fail to edit out an artifact, like bleed through, that the software, that the ProbGen software doesn't model itself. So here is a listing of the mixtures that we used in our first validation. And these are the data from our publication on the validation of our probabilistic genotyping system. And then for our second validation, we used a sort of a similar number of mixtures, but at this time, we decided not to go up to 5-person mixtures. That honestly was given the deadline for set by the FBI for implementing the expanded CODIS core loci, we ran up against a time crunch for that, and so we decided at that point to limit our analysis to 4-person mixtures. In fact, with our initial application, we would interpret in our ProbGen system 5-person mixtures that were conditioned on a contributor. So we found that that particular scenario, we didn't really encounter that very frequently in Casework, so decided not to include that in the next validation. But in our future validations, that's something that I would like to include contributor numbers that do go higher than what we have done at this point. So sensitivity and specificity are two additional topics that are included in both the developmental and the internal validation. Sensitivity, now, sometimes we think of sensitivity in terms of PCR, sensitivity of detection. What's sort of a similar phenomenon when you think of a probabilistic genotyping system? It's the ability of the system to reliably determine the presence of the contributor's DNA, so this is a true contributor, over a broad range of evidentiary typing results to include mixtures and low-level DNA quantities. Specificity, on the other hand, entails studies to evaluate the ability of the system to provide reliable results for non-contributors over a broad variety of evidentiary typing results as well. So to that end, here are the mixtures that I showed you a couple slides ago, showing you in the second to last column the number of H1 true tests. So this is the sensitivity testing. So, for example, 106 mixtures, each having 2 contributors, would give us 212 tests. It's generally how this panned out. And then we took a database of genotypes that were artificially created to have a large number of them and tested them as well. These are non-contributors. So you can see there that a very large number of tests were done for both sensitivity and specificity testing. And then I'm going to show you on the next slide when we used our expanded CODIS core loci amplification kit. This is the number of contributor and non-contributor tests that we tested. Now, what SWGDAM says about this, or what we're actually looking for, is for the specificity testing on the left, we're testing non-contributors, and then we're going to see if LR is greater than 1 would be generated for these non-contributors. And for the true contributors for sensitivity testing, our LR is less than 1 generated. So ideally, I'm going to set this up sort of as a plot here, going from 0 to 1. This is where we would hope to find the results for all of our non-contributors, that is supporting HD, and for HP support for our true contributors, we want to see them on this side of the plot. So first, the non-contributor testing. And I want to make the point, if you simulate enough non-contributor profiles, you will surely find one eventually that returns inclusionary support, okay? So we've got a mixture of however many contributors, and if we are randomly generating genotypes or testing enough known contributors, we will eventually, in the population of artificial or true individuals testing more and more and more genotypes, find one that will fit in the mixture. So what we have here, this is the result from our expanded locus kit of non-contributor tests. We would ideally have them all down here with an LR of 0. We saw them all along the scale, and this, by the way, is a compilation of data from our 2 to 4 person mixtures, and we did have some that fell in the uninformative range with an LR of 1, and then some that provided support for HP, with a maximum false HP support for a non-contributor being at 95. This, though, is not the expanded locus kit. This is an example of the kit we used before that. And this is one where a non-contributor gave an HPD LR of 862. And what we have here is the minor contributor peaks, or positions where this contributor, matched the minor, shown in green, where it was masked by the major, would be shown in purple, where it could be considered dropped, or in a stutter positions indicated, and so forth. So this is a particular non-contributor that just happened to fit in this mixture. And the only questionable position was here, a peak that was modeled at 9% stutter. So this is how it can happen that a non-contributor can match a profile. And this is, in fact, nothing really new to probabilistic genotyping. What kinds of data results in an adventitious HP support for a non-contributor? Well, first of all, what our data shows is that these are generally contributors that are less than 50 picograms of the total. And you'll see here that most of the HPDLRs are under 10, or certainly under 100, with just this one exception, which is the 862 that you saw a moment ago. They have extensive dropout and a low average peak height. If you could see the scale, you might ask, how can you have an average peak height of 20 or 0 and so forth? The reason that you can have that in this average, if an allele is dropped, it was assigned for this analysis a height of half of the analytical threshold. So that was what drove these values down. And it indicates here, based on this plot, that we have a lot of dropout and low average peak height, and 0 to 4 obligate alleles. So this is a non-contributor whose peaks are not just standing there unshared for you to see in the profile. So for the most part, very few obligate alleles. Now if we switch over to contributor testing, this is a sensitivity testing of known contributors, again, 2 to 4 person mixture. So this is a great result over here for true contributors returning high LRs, and across the range of all of our 2 to 4 person mixtures, we see a spread of data. And if we look at what's happening here, all of our mixtures we ran through the program, but upon analysis of the data, looking at those profiles, these ones in this range are actually ones which we, based on our casework procedures, would not have actually subjected to probabilistic genotyping, because our policy, we do a visual interpretation of the data first, and if there is an obvious exclusion, we will declare an exclusion outright. without even running ProbGen in certain instances. So in my reviewing the data, I would have just excluded these because they're very, very low-level contributors that just basically weren't detected in the profile. So then if we were to focus on the ones where I wasn't sure, or the ones I would have run it through the ProbGen program, it basically 1 over 826 is the maximum, is the going in this direction, the lowest LR that was obtained for a true contributor that would have been run through our ProbGen program. And what kinds of data results in an incorrect HD support for a known contributor? Well, we didn't have any instances in 2-person mixtures, but in our 3- and 4-person mixtures, less than 50 picograms, extensive dropout. Look how many, this is maximum average peak height of 0 or near 0. So extensive dropout and very, very few obligate alleles. So these are just exceedingly low levels or low detection of these contributors. And this is exactly what we might expect the software to do in these situations. So here's both the sensitivity and specificity data plotted together. And that is something that we are going to be focusing on again when we come back in our next session to talk about the SWGDAM guidelines for what to do with these data. But summarizing our data, here we have the contributor expectations, where yes, there was a portion of our data that gave an LR of 1, but 60.7% of our known contributors gave support for HP, and then 35% of them gave support for HD. For our non-contributors, 99.62% gave support for HD or an outright exclusion. We had a small proportion returning an unformative LR, and then this percent, 0.09% of our, I think, our tens of thousands, I don't remember the number, of non-contributor non-contributor tests returned support for HP. One thing to point out, and the reason I gave it a star there, is to make the point that you shouldn't misconstrue this as some sort of an error rate. Your non-contributors giving HP support is actually not considered an error. So despite the fact that in the guidelines, we do say sensitivity studies should demonstrate the potential of type 1 errors, which is an incorrect rejection of a true hypothesis, or specificity demonstrates the potential of type 2 errors, which is a failure to reject a false hypothesis. These are really not, in our view, considered errors. Errors of the probabilistic genotyping system. And the reason is as follows. Dr. Buckleton, in one of his testimonies, stated, "We've done some massive tests of false donors, hundreds of millions, and we occasionally get a weak indication of inclusion at exactly the error rate expected from genetic principles. So they're not false positives by the software. They're false positives because we've, by doing so many, actually replicated someone who is genetically similar to a true donor. So if you create a genotype that should be included, it is. That's what the software does. Ian Evatt also stated, there is no doubt that performance must be expressed in terms of the LR given in HD. An LR greater than 1 when HD is true is not an error. So again, if we're providing to the software a genotype that matches the mixture, then the software is going to return some level of support for that. So let's talk about another category of false exclusions. These are ones that we included this in our validation paper, and they're instances where at a single locus, an LR of 0 was returned for a known contributor test, while the other loci in the profiles returned intuitively correct LRs that were greater than 1. So this is not the case where we would look at the profile and say, oh, yes, there is a result at that locus that is exclusionary, and truly that person should be excluded. This is looking at the profile and not agreeing at this particular locus that there is an outright exclusion. Well, some of them occurred in saturated 3 or 4-person mixtures. One occurred in a 3-to-1-1 mixture. I'm going to show you in the next slide where there was an unresolved minor allele that differed in size from a major allele by 1 base pair. And then the other instance was 4 mixture replicates that returned an LR of 0 as a very infrequent result of the statistical sampling process, the MCMC, that just occurred when the probability space that includes the true genotype happened to not be sampled. Most, but not all, produced a non-zero LR for the affected locus upon reanalysis. So this speaks to the point that when we have tested our known contributors, it's possible that the software, it's not returning an error, but it's returning an unintuitive result that the diagnostics that come along with the software show you that something happened in this particular run and I need to run it again. Okay, so we put these in our validation paper to support the fact that sometimes a probabilistic genotyping analysis is warranted. Sometimes you may require a replicate injection or a replicate amplification or some reanalysis. And what is said about probabilistic genotyping along these lines is that you need to correct the biology before you attempt to correct the probabilistic result if you see that something is unintuitive with what's going on. So here's the data from the paper. This mixture A we can talk about, for example. This is the initial run where at SO1 we have LRs greater than 1, and then we have this 0 here, and then if we actually had a replicate amplification of that where the alleles were resolved. And so this gave an appropriate LR that was intuitively correct for how we would have interpreted it. If you can do a replicate PCR, that's great. If you can't, we know there's something is biologically incorrect at this locus based on a review of the profile, and it is appropriate to omit the locus from the ProbGen analysis in that case. And here on the top panel is where this particular 10 allele, this is the 3-person mixture, where the 10 allele was clearly not resolved and something was clearly wrong at electrophoresis. So false exclusionary results for known contributors, these are not errors of the software. They also have occurred due to human error. So for example, we might have submitted or put into the software the wrong reference, or we might have failed to edit out a bleed-through peak. When the input data are corrected and reanalyzed, the correct results are returned. And then I mentioned the incomplete resolution. And then another issue has to do with apparent number of contributors is less than the true number of contributors. Okay, so we may know that we have created a 4-person mixture. Our 4-person mixtures, if we were to analyze them according to casework procedures, some of them with extensive dropout or failure to detect a contributor were interpreted as an apparent 1, 2, 3, or 4-person mixture due to allele masking, dropped alleles, undetectable contributor, minor contributor, modeled as stutter, and so forth. Now I want to move on to the analysis of replicate amplifications. So I was having a discussion with someone a couple weeks ago, and as we were talking about some of these topics, his response to many things was, well, just re-PCR it. And I finally figured out why our approaches were a little different. their final extract volume is 50 microliters. We, since we have moved to a solid phase extraction process, we don't have problems as we did in the past with inhibition, or we encounter it much less frequently in the past. We decided to start speed vacuuming most of our evidence types so that our extracts are in a total of volume, so that the entire extract could be used in a PCR if we had a low template sample. So for some of these challenging ones, we have nothing to go back to since we've concentrated the extract in that way. But other laboratories have a higher volume than us, and replicate amplifications would be a consideration. So let me show you, there's definitely a benefit to some data for analyzing replicates. So what we're looking at here, this is a log HPD LR scale, where we're looking at the results for a single amplification, or now what we're talking about is this software program allows you to take the results from replicate amplifications of the same exact extract and analyze them together in one run of the software. So are those replicate amps analyzed together any better than the single? And the answer, just intuitively, would seem yes. And it's shown in the data here with 52% of the data. And by the way, these are two-person mixtures, and the squares represent three-person mixtures. So 2- and 3-person mixtures, and the white represents one order of magnitude difference. So 52% of them showed a greater than 1 order of magnitude increase, and just 3.4% showed a decrease. So really not a significant number here. But then let's talk about what's going on with the non-contributors to 2- and 3-person mixtures. So we're going to zoom in here. But first, showing the data that's here, we have incorrect support for HD. So that would be the squares with the LRs less than 1 that are here. All but one, that would be all but this one, improved when we ran the replicate analysis. But keep in mind that these are actually the worst of the worst 3-person mixtures that we had. So some of them in a single amplification process produced values less than this line. So that's single PCR HD supporting. But for the replicate, they showed improvement above the line, or either improved here or gave an LR value greater than 1 on that scale. Okay, so now talking about the non-contributors that are shown as red. First of all, non-contributor replicates were only analyzed if the first analysis provided incorrect for HP. So those are these triangles. So that's why you don't see a lot of stuff over here, okay? So PG analysis of the AMP replicates returned correct support for HD for 61%. The remainder, the impact of replicate analysis on the LR was less than 1 order of magnitude. So that would be what we're seeing here in this white zone around the line, okay? So replication, again, improved the results. But oftentimes, the decision, there are two ways to look at it. Are we talking about taking, and the next 2 slides are going to be for our expanded locus kit. So the question is, should we do 1 PCR or 2 replicate PCRs with a concurrent PG analysis? So like we saw with the previous AMP kit, we have an improvement in the data. But oftentimes the analysis is, the issue is, should I have an extract. Should I amplify the entire thing with 1 PCR? Or should I split it and do 2 half template amount PCRs in order to benefit from a replicate analysis? And the answer to that is amplify the full thing in one PCR. And the reason, I think, is because the ProbGen software does a really good job of modeling dropout and dealing with stochastic effects. So the data are shown here, where we're looking at single source 2, 3, and 4 contributor mixtures, where we're talking about half X replicates, so splitting my sample in half in order to replicate it, or just taking the whole thing as one. It's actually not a replicate. And everything supported. There wasn't a single instance of improving performance by splitting an extract in half in order to replicate the amplification for concurrent PG analysis. And then here we just got some individual sample results where we're looking at replicates, where you see a star, that's a single, and a circle means replicates were performed. So you can see how the replicates increase the LR on the scale here. This is increasing template amounts. You could also look at going from a higher to a half template amount and seeing there's really no benefit, again, as we saw in the other slide. The next topic I want to move to has to do with using alleles under the analytical threshold to estimate the number of contributors. And that we have seen can prevent a false exclusion of a low-level contributor. Here's a profile. This is a 62 picogram single source example. Now, this I think would be modeled really well in the ProbGen software, but imagine if we had additional contributors to this mixture and we were trying to figure out what the number of contributors to that mixture is. So I've shown here with the black triangles, there are some alleles that fall under our analytical threshold. And I bet you as well, if you were able to sit at your analysis software and zoom in on those results, you might have a pretty good feeling for the fact that the baseline is really clean and very low in some samples, and that you could be confident that, especially if you were to reanalyze this at a lower analytical threshold, that you've got some data here that might be reasonable to use in estimating the number of contributors to the mixture. Now, we wouldn't want to do that in a profile like this, but if there was ambiguity or difficulty in calling the contributors, and you had contributors that were right around the analytical threshold and below. And one of the reasons to do that, I could try to demonstrate that with these cartoons. So we have here donor 1 that has a genotype 2.5. And here's donor 2 that has the genotype 1.3. And we have it, it's one of its 2 peaks below the analytical threshold. We have another minor contributor that has the genotype 2.3. So let's mix them. And those alleles are stacked. So the shared peaks, we're going to represent purple as a mix of blue and red, and blue and yellow are the shared green peaks. So that's the result, okay? So I think what I'm representing there is the stutter peak, being modeled as stutter. And if we didn't consider those 2 peaks below the analytical threshold, I think it would be reasonable to say that this might be a 2-person mixture. Now, obviously, you're going to look at all the other loci and helping to make this determination, but sometimes that data under the analytical threshold might have been useful to you. Because this, if we were to call it a 2-person mixture, what might happen? Well, let's see. Donor 1, who's a 2.5, I would think that one is included. Donor 2, which is a 1.3, would be considered possible as a minor contributor with some dropout. But then your third donor, Mr. 2.6, who's actually shared alleles with the major contributor, would be excluded because no one can explain, the first two don't actually explain, he doesn't explain the presence of that one peak. So had we looked under the analytical threshold and decided that this could have been a 3-person mixture, then that might be something that would be suitable to test during your validation phase to see if there are data types for which you might think it's appropriate to be guided by data under the analytical threshold, not for doing the ProbGen analysis, but just for help in assigning the number of contributors to a mixture. Hypothesis testing, I'm just going to mention because I don't have any data in the presentation about it because it's going to be the topic of one of our upcoming lectures and another module. But the hypotheses used for evaluation of casework profiles, they can have a significant impact on the results obtained. So as part of the validation process, you should develop various hypotheses with contributors and non-contributors and test more than one set of hypotheses that will help guide you in developing your laboratory's procedures for how you are going to formulate hypotheses. So for example, the one scenario would be if you have a mixture and there are 2 subjects in the case, you might decide that you might want to test as part of your validation, what do the results look like if you were to test person of interest 1 and 2 concurrently versus the hypothesis, the prosecution hypothesis of person of interest 1 plus an unknown, or person of interest 2 plus an unknown. And you might find some situations as well where one of those in the mixture, or one of those persons of interest might be a strong contributor to the mixture and the other might be a non-contributor. So it would be interesting to see how the software handles that scenario with a strong contributor with a lot of their DNA present together with a non-contributor, what the LR looks like in that situation. And as well, something that we do a fair amount as well in casework, which is conditioning or assuming the presence of a person's DNA in certain evidence items. Moving on to the topic of contributor numbers. I'm not sure if Duncan's going to be talking about this as part of his talk later on, but this is a plot from one of his papers with Joe Bright, where they demonstrated for a very, very large data set that underestimating the number of contributors tends to have either no effect on the LR or to exclude known contributors. So you might have heard it said that underestimating number of contributors is either correct. I'm sorry, that doesn't make sense. It's either good or it's conservative. I don't know if I said that right, but essentially there is little ill effect for underestimating the number of contributors to a mixture in the ProbGen world. This was an issue when we were calculating CPIs because it informed on the possibility of missing data. So we needed to know how to be able to estimate accurately what the number of contributors was, and it's less of significant now that we've moved to probabilistic genotyping. Now, let's see, setting contributor number. I guess I'm going back to this topic that we spoke about a moment ago, setting contributor number based on alleles above the AT or above and below the AT. And this is kind of a complex graph that I'm going to have to step through. This is 3-person mixture. So the first thing we're going to do is divide the plot into 4 quadrants. And then this would be an instance where, first of all, we're looking at establishing contributor numbers, just looking at data above the analytical threshold and calculating the LR, and then comparing that to using data above and below the LR. And in this test, we're going to be evaluating both non-contributors and contributors. So here is our line of equivalency where we see that both are correct for contributors and both are correct for non-contributors. Now, what we see, though, is that lower exclusionary potential in this situation. So basically, get my cursor to work. If we are using data above the analytical threshold only, we get a low value here versus above, and we get a larger value than if we were using data above and below. Okay, so it's closer to that line there. So we see that we have a lower exclusionary potential since there's more matching opportunity with the higher contributor number. So that's something that just makes intuitive sense. If we, the data that we see here, though, that for non-contributors is generating LRs that are greater than 1, if we were to use data above the analytical threshold, we see we've got some points that are right on, it's shown as 30, but these are really LRs of 0, or they are over here in this area. To zoom in on that, though, one point to make is that these are LRs that are very close to 1, and the HPD is likely to be closer to 1 as well. What we see, though, for true contributors is that if we were to only use above the analytical threshold, we've got exclusionary results, whereas it's greater than 1 if we were to look above and below. Okay, so that's a good thing. And that is supported as well for the true contributors and for 4-person mixtures, the same sorts of results. If we were to incorrectly assume the number of contributors as n+1, this is one of the tests that is also recommended by SWGDAM to test both correct and incorrect contributor numbers. So we're going to look at the number +1 as well as the number -1, and important that you assess both known contributors and non-contributors. So if we assume too many contributors in this fashion, what we see is that the effect, looking at single source, 2 or 3-person mixtures, is that typically the difference is less than 1 order of magnitude, but it may lower the LR for a trace contributor, and that occurred in 13% of our data. So that is for true contributors. And then with non-contributors, what happens is there are more low-level adventitious links with non-contributors and far fewer outright exclusions of LR of 0 as expected. But the vast majority of the results, more than 98%, still resulted in an LR supporting HD. Incorrect assumption of n-1 contributors. Okay, now this can be difficult to assess because the probabilistic software may not proceed with analysis if the number of alleles in the profile cannot be explained by the number of contributors that you've entered. For example, if you've said it's a 2-person mixture and you have 5 alleles at a locus, the analysis is just going to stop, okay, or at least for the program that we tested. So the way that we tested this was we added in artificially a 3rd contributor to 2-person mixtures. So this you could think of essentially as being 2 parents, the genotype represented in blue and the genotype represented in red, and a child of these parents represented as the yellow as a trace contributor to the mixture. And then we're just going to add those heights together. So that is the 3-person mixture that we have artificially created by adding a 3rd contributor to a real 2-person mixture. And then this 3-person artificial mixture, we would test it if it were interpreted as it could be as a 2-person mixture. We did that sort of addition as the 3rd contributor to all of the loci in the mixture, and then test the known contributors. And what we found was the LR of the major and minor contributor was not affected by the addition of a 3rd contributor at any of the average heights. that we tested. Now, obviously, if you're going to be adding more and more and more of this third contributor, it's going to throw the heights off of the peak such that the heterozygote balance is outside of expectations, and it would have an effect on the LR. But at these low levels of mounts, it would be reasonable for you to still think that this is a two-person mixture rather than a three-person mixture. The software was not affected by that at all. So that is all of the data and information that I have to share with you on validation of probabilistic genotyping systems. So just some things, a couple of concluding points to think about, and that is that mixed DNA profiles containing more contributors are not unreliable. They are just less informative with respect to potential contributors, and they return less discriminatory LRs. The more complex your mixtures, it may be the case that your LRs are approaching 1, and that in itself does not mean that those mixtures are necessarily unreliable. We need to test them during our validation. As the number of contributors increases, an increased number of potential genotype sets can explain the data, resulting in an overall reduction of the weights assigned to each set. And what does that do? It pushes the LR towards 1. So when you think about that, increasing your contributor number is basically spreading out the weight. This is all stuff that just makes logical sense. And it's what we see when we validate our system by running the test. So my thoughts leaving the topic is that don't restrict your usage of probabilistic genotyping software based on what you understand to be the limitations of a binary approach. We all have approached these validations thinking certain things about mixture interpretation based on our years of experience of analyzing mixture, applying thresholds and stutter guidelines and peak height ratio expectations, and thinking about allele sharing and dropout and so forth. But it is pretty amazing to go ahead and try more and more complex things and see how the software is able to handle it. Because when you ask the question, the mixtures are too complex for what? And often the answer could be too complex for my brain to process all the multifarious types of bits of information and things that go into the interpretation. But that is exactly how the computer systems are designed to do that. So create the mixtures that are necessary to fully explore the software and run the tests as part of your internal validation to inform your policy and your decisions. So that is everything that I have for you today. And I want to thank a whole number of people who have helped with our validation studies and our process of implementing and using probabilistic genotyping at the FBI. So many people there have helped us on this path. So thank you all for your attention today. And what I would like to do at this point, take a look at the clock. Let us take, it's 1:41. Let's take a 10-minute break and come back around 1:50. So thanks very much for your attention today. If there's any questions that have come in during my talk, I am going to review them and I'll add them to our question and answer session at the end of the session. So we'll take a break till 1:50 and come back with Dr. Noel. Thanks very much. Okay, welcome back everyone, and thanks for your attention. We're ready to resume again. And at this point, I would like to reintroduce Dr. Sarah Noel and invite her to begin sharing her presentation.
Okay, so I am starting my screen sharing. Hope everyone can see it properly. So good afternoon. I am going to talk about brothers and DNA mixtures, but first I will do just a quick word of presentation. I am a DNA analyst at the SWGDAM, which is located in Montreal in the province of Quebec, Canada. The SWGDAM is a government lab, so our main focus is to do casework. We do about 6,000 cases per year, and our DNA section is composed of 36 DNA analysts. We started the transition to probabilistic genotyping at the end of 2015 when we acquired the software StarMix. And we have been using it in casework for a little bit more than a year. But prior to that, we worked on the validation for about 2 years. And a big part of our validation was to do a large specificity study with a database of 300,000 non-contributors. So here is the reference on that part of the validation that was recently published, if any of you are interested. But today I want to talk about some additional validation that we did a couple of months after the implementation of StarMix for casework, when we got interested by the question of relatives, more specifically the brother defense. So to explain briefly what is commonly called the brother defense, I will take a simple example. Let's say a murder was committed and the knife was found at the crime scene. The handle is swabbed and the genetic profile is obtained and it matches a suspect, and we will call him Mr. Blue. And Mr. Blue is arrested. And then Mr. Blue says, well, it's not my DNA that is on the handle of the knife, it's my brother's. So that is the brother defense. More formally, it's when H1 is that DNA comes from Mr. Blue, and H2 is that the DNA rather comes from a non-specified brother of Mr. Blue. And the brother defense actually has a scientific basis because we know that DNA profiles from relatives are more similar to each other than profiles of unrelated. Here is a chart showing the total number of shared alleles between pairs of unrelated in black and pairs of sibling in red for global filer. We see that most pairs of unrelated individuals share between 8 to 18 alleles, while most pairs of siblings share between 22 to 32 alleles out of 42. So because DNA profiles of siblings are similar, they share more alleles on average than unrelated pairs. There is a greater possibility that a sibling of Mr. Blue could also match the profile that was obtained from the knife. At the beginning of DNA analysis, the beginning of STR analysis, very few loci were used, so the probability that a brother of the person of interest could not be excluded was rather high. But as more and more loci were added to the standard DNA analysis, that probability decreased for single source profiles. However, over the years and with the introduction of probabilistic genotyping, forensic community has started to work with more and more complex mixtures. And so the question of the brother defense is highly relevant in the context of those complex mixtures. And we will illustrate that with the example of Mr. Blue and the knife handle. So Mr. Blue was arrested, he was genotyped, and he is a TEM12 at a given genetic site. Now imagine that a single source profile was obtained from the knife handle, and there is a TEM12. So for a sibling of Mr. Blue to match with this evidence, there is only one option. The sibling also needs to be 1012 because we have a single source profile. Now, if instead of a single source, we have a 3-person mixture that are in similar proportion, the evidence could look like that. Now, for a sibling of Mr. Blue to be included as a possible contributor, there are many, many genotypes that are possible. So how should we proceed when the brother defense is proposed in a case? For example, if the defense attorney says something like, what if I tell you that DNA on the knife handle comes from the brother of Mr. Blue? Do you think it's possible? I think it was Peter Gill who mentioned in response to a question a couple of weeks ago on the webinar that the best way to answer this question is to obtain the brother's profile and to compare it to the evidence. However, this is not always possible for a variety of reasons. So can we evaluate this hypothesis anyway when we don't have the DNA profile of the sibling? The answer is yes. The best option will always be to genotype the brother, but if it's not possible, then there are some calculations that can be made. And we are going to look a little bit at those calculations considering relatives, namely the unified LR and the sibling LR. But first, we will review very quickly the standard LR. So in the standard likelihood ratio, the propositions considered are either the DNA comes from the person of interest, Mr. Blue, or from an unknown individual that comes from a population of unrelated individual. This is why the little men here are in gray, because they are unrelated to Mr. Blue. In the unified LR, H2 is that DNA comes from an unknown individual that could be unrelated or related to the person of interest. So you see the little men that are in blue here, they are related to Mr. Blue. They can be a brother, a father, so they are from the same family. The unified likelihood ratio considers that the population is a mix of related and unrelated individuals. And then you have the sibling likelihood ratio, where H2 is that DNA comes from a non-typed brother, an individual who is taken from a population of possible brothers of the POI. So those three LRs are calculated in StarMix. I should also say that StarMix calculate likelihood ratios similar to the sibling LR, but for other relationships, such as a parent, cousin, uncle, etc. The calculation of the probability that the brother of the person of interest could also match the evidence is a question that has sparked the interest of mathematicians, statisticians, and forensic biologists for many years. And they have developed equations and models that are more and more refined, including now allowances for peak heights, for example, or population structure. And these are some examples here, and this is in no way an exhaustive list, as many other important books and papers were published on this question and have established a mathematical framework that is used to calculate the likelihood ratio that we have seen on the previous slide, the unified LR and the sibling LR. So mathematicians, statisticians have provided us with ways to calculate those probabilities, very nice equations. However, equations are not easy to grasp for everyone. So some DNA analysts may be comfortable with them, but others not that much. And when it comes to the jury, most of them are not scientists, and mathematical explanations can be very scary for them. So the way I see it, equations are a little bit like a music sheet. If you haven't studied music, if you are not a musician, you look at the music sheet and you only see a jumble of black marks. But if any of you are musicians, you may have recognized here the theme music for Game of Thrones. So with the objective of adding another tool to the toolbox of DNA analysts and to better understand the broader questions in the context of DNA mixtures, we decided to do an empirical study. As I said in the introduction, that part of our validation came a couple of months after we started using StarMix and Casework. And we wanted to investigate what is the risk of accidentally obtaining a likelihood ratio that would support inclusion for a sibling of the true contributor in a DNA mixture. So we are going to take a quick look at the steps that we took for this empirical study, and then I will briefly discuss each of those steps. First, we selected 10 DNA mixtures. They were then amplified. We deconvoluted them with StarMix. We then targeted a person of interest in each of those mixtures. We built a database of brothers for those persons of interest. And finally, we computed the likelihood ratio of the brothers. So step one, the first step was to select 10 DNA mixtures. And we wanted to have a variety of mixtures that would represent the range of results that we have in Casework. So we selected different DNA quantities, different number of contributors, and also different proportions. Here you can see the 4 high template mixtures that we had, where all alleles were above the stochastic threshold. You can see the DNA quantities there. They were from 2, 3, or 4 persons, and those high templates mixtures are identified as H1 to H4. We also had 6 low template mixtures where at least one contributor had low DNA quantities and alleles that fell under the stochastic threshold. And in our lab, we start to see dropout at about 100 picograms. So you see those low-level contributors indicated in red in the table here. And as soon as there was one low-level contributor, the whole mixture was considered as a low template. Those low template mixtures were again composed of 2 to 4 persons, and they are identified as L1 to L6. Finally, different configurations were chosen. We had some major minor with 1, 2, or 3 minors. We had contributors that were more or less in equal proportions. And we also had some mixtures where contributors were more in crescendo proportion. Step 2, amplification. Each DNA mixture was amplified once, and we are using the identifier plus kit. Because we are in Canada, we had no obligation to switch to a megaplex in 2017, so that is why we are still using ID+. And samples were run on a 3500 CE instrument. Step 3, deconvolution. As I said earlier, we are using StarMix, so samples were deconvoluted in StarMix, and we are using version 2.5. After the deconvolution, the proportion of contributors and diagnostics were checked to be conform to our expectations. Step 4, we targeted the person of interest. So in each of the different mixtures, one contributor was targeted as the person of interest, our Mr. Blue, and so he is indicated in blue in the table. In some mixtures, it was contributor 1, which is in the largest proportion. Sometimes we choose the second or third contributor. So depending on the mixture, the DNA quantity of the person of interest that was targeted was between 50 and 700 picograms. Step 5, building databases of brothers. The databases were created with R and the package DNA profiles. I will very briefly explain how the databases were built. Using the genotype of the person of interest here, using the genotype of our Mr. Blue, and also using the formulas that Mike Coble presented a couple of weeks ago with the probability of having 0, 1, or 2 alleles that are identical by descent between brothers. And also using the allelic frequencies of the population, it is possible to simulate the genotypes of brothers to Mr. Blue. So for each mixture, we created 10,000 brothers of the person of interest. So a quarter of those brothers will have 0 allele identical by descent with Mr. Blue. Half of them will have 1 allele IBD, and the last quarter of them will have 2 alleles IBD. Of course, most people don't have 10,000 brothers, but in order to explore the wide range of variability that can exist among brothers, we wanted to have very large databases. Finally, each deconvoluted mixture was compared to the database of 10,000 DNA profiles of brothers, so that 10,000 likelihood ratios were obtained for each mixture. Okay, let's look at the results. Here we have a chart showing the likelihood ratios of the brothers of the person of interest in the DNA mixtures. For now, I am showing you just a part of the chart to explain it. On the x-axis, you have the first mixture, H1, which was a high template mixture, so that's why the data points are in green. On the y-axis, you have the likelihood ratio on a logarithmic scale. So LR of 0 was attributed log LR of -30. LR of 1, which is neutral, equals log LR of 0. So any LR larger than 1 is supporting inclusion, and any LR smaller than 1 is supporting exclusion. And because here we are comparing non-contributors, we are comparing brothers of the true contributor, who is Mr. Blue, most data points are expected to be in that zone, to be supporting the exclusion of the brothers because they are not, in fact, contributors to the mixture. The black square here represents the median. Here the median is at 0, meaning that most siblings had a likelihood ratio of 0. Other siblings had a likelihood ratio between approximately 10 to the -15 and close to 10 to the 10, which is 10 billion. We use the database of 10,000 brothers, so there are actually 10,000 data points here. We cannot see them all because there is some superposition. And also, most of them are hidden under the black square of the median. Because the median is at 0, it means that underneath the black square, there are at least 5,000 data points. Here you have the results for the other 3 high template mixtures. We see that H2 and H3 are very similar to H1 with the median at 0. And H4 has a slightly different distribution with likelihood ratios that are in general a little bit larger, but the median is still under 1, supporting the exclusion of siblings. And here you have the results also for the low template mixtures, L1 to L6, they are in red. And the distribution is a bit different. We can see that the medians are close to 1. And we can imagine that this is because of the presence of a low-level contributor. So let's look at a simple example. Imagine you have a major minor with 1 allele of the minor that is barely above the analytical threshold. So we know there is not much DNA from this minor contributor. So what are the possible genotypes for this minor contributor? we know he needs to have the allele 12, so it could be a 10-12 or a 12-12. But because it's totally possible that its second allele dropped out, any genotype with the allele 12 would be possible. So that makes it possible for a larger number of siblings to not be clearly excluded. And that may explain why there are no red data points at 0 for low template mixture. Globally, we had 36% of the brothers who had a likelihood ratio larger than 1, so that supported their inclusion. Some of them were pretty close to 1, but others were quite large. So we will look a little bit more in detail at the different degrees of support that were obtained. And I will make a small parenthesis here to talk about the different ways to express the degree of support. And one of them is to use verbal qualifiers. So here you have the verbal scale recommended by the SWGDAM to report likelihood ratio. LR of 1 is uninformative. Between 2 and 99, you have limited support. 100 to almost 10,000 is moderate support. 10,000 to almost 1 million is strong support. And then over 1 million, you have very strong support. And the color code that I have chosen here may seem counterintuitive, but as I mentioned previously, in this empirical study, we're comparing non-contributors. So most of them are not expected to fall in the strong or very strong support categories. In our lab, we do not use a verbal scale, but rather a visual scale that looks like that. I am not sure that you can read the small fonts, so here you have a little bit bigger. So any likelihood ratio smaller than 1 is an exclusion. We have an inconclusive zone from 1 to 1,000. And then you have kind of an increasing volume representation showing the increasing strength of DNA evidence. So we felt that it was important to find a way to convey the strength of DNA evidence that is obtained. But we were not satisfied with the option of a verbal scale. So we created this visual scale that is sent along with every report. For comparative purposes, I have indicated here the color equivalents for the SWGDAM verbal scale, and we will use those categories for the rest of the presentation. So this is the end of the parenthesis. Let's get back to results. Here you have a bar chart showing the likelihood ratio of brothers separated by degree of support. Once again, I'm showing you only a part of the chart first. On the x-axis, you have the 3 high template mixtures, H1 to H3. On the y-axis, you have the proportion of the likelihood ratios that were in each of the SWGDAM verbal scale categories. I am using the same color code as the previous slide, except that the blue was added to represent exclusions. So in green, you have limited support for inclusion, and then the more you go towards warm colors, such as orange and red, the degree of support increases, which is not what is expected for our non-contributors. So for H1 to H3, almost all of the siblings had a likelihood ratio smaller than 1, so they fall in the blue category of exclusion. And a very small proportion were in other categories, but we don't really see here because it's very small. If we look at H4, we see that 86% of the 10,000 siblings had a likelihood ratio smaller than 1. About 11% had a likelihood ratio with limited support, so between 2 and 99. Overall, the largest category for high template mixtures was exclusion. And here you have the results for the 6 low template mixtures as well. There are a couple of things to notice on this chart. The first thing is that for all DNA mixtures, most brothers are either excluded, so they are in blue, or have limited support for inclusion, so they are in green. The second thing to notice is that a likelihood ratio larger than 1 million were obtained for all mixture types, so for both high and low templates. I'm not sure you can really see, but there's a tiny slice of red here at the top of H4, except I think H4. So representing likelihood ratio that fall in the very strong support categories. And very strong likelihood ratios were always observed in very small proportions. It was always less than 1% of the siblings. And the maximum likelihood ratio observed for each mixture was pretty large for both high and low template mixtures. Up to billions and more were obtained. So we took a closer look at those siblings with very large likelihood ratio. And we counted the number of alleles that are shared between those siblings with likelihood ratios in the order of billions, and we see that most of them had 24 to 27 alleles in common with the person of interest. Here you have the same chart that I showed you at the beginning of the presentation, but for identifiable plus this time. And you see the total number of shared alleles for pairs of unrelated in the broken line and pairs of sibling in the full line. And you see that the number of alleles shared with Mr. Blue for these sibling with a very, very large LR is in the little red zone here. So this is completely to the right of the distribution curve. If we go back to that chart, the other thing to notice is that as we have seen on the scatter plot a couple slides ago, low template mixtures led to more likelihood ratios larger than 1 for siblings when all the support for inclusion categories are pulled together. So here I have indicated the percentage of the 10,000 brothers having a support for inclusion. For high template mixture, it ranges from almost 0 to 14%. And for low template, it is mostly from 49 to 87%, with the exception of L3, which has only 2% of sibling with a likelihood ratio larger than 1. So we see that the specificity is not as good for those low template mixtures. So is there something we can do? In our lab, we routinely use replicates for low-level samples. As Dr. Moretti says, different labs do different things. We are a lab that use a total volume of extracted DNA of 50 microliters, so we do use replicates. And as it's indicated in the StarMix manual, when possible, we should always try to improve the quality of the data biologically. It could be by concentration, by speed vac, or with, for example, PCR replicates. So we decided to include some replicates in our empirical study. Each mixture was amplified a second time in exactly the same conditions, and a new deconvolution was done using the two results together in StarMix. The deconvolution was then compared to a reduced database that contained only the siblings who had a likelihood ratio that was larger than 1 with only 1 amplification. So for each mixture, the siblings who were excluded with 1 result, those that fell in the blue category, were removed from the database. And only siblings for whom the likelihood ratio supported inclusion were kept in the database. because we wanted to assess what would be the effect of using a replicate for the likelihood ratio of those siblings. So depending on the mixture, the sizes of these reduced database were between 50 and 8,700. So here are the results. We are going to first look at H1, the first high template mixture. This pie chart represents the proportion of siblings who had a likelihood ratio that fell in the different categories of support for inclusion and using only one amplification. And the bottom pie chart shows what happens when you use two amplification, when you have two results. We see that a big proportion of the siblings that had the support for inclusion in different categories, they are now excluded, they are in blue. Another thing that we can see is that the red slice increased a bit when we used two replicates. In fact, there was only 15 siblings with a likelihood ratio larger than 1 when using one amplification, and one of them was larger than 1 million, so that's the small red slice. But when a second amplification was done, a second sibling had a likelihood ratio larger than 1 million, and that sibling shared 25 alleles out of 30 with the person of interest. So that explained the large LR. Here you have the complete results, and we will not look at each of the pie charts separately, but we see that there is a kind of a general pattern that the bottom pie charts have more cool colors. We see more blue and green and very small orange or red slices. For some mixtures, doing a replicate really reduced the number of siblings with a likelihood ratio supporting inclusion. For other mixtures, the effect was not so radical. So generally speaking, when you do a replicate, you have more information, and that increases the specificity. So more non-contributors are excluded. And that brings us to the conclusion. So in total, we had 10 mixtures. Each of them was compared to a database of 10,000 brothers, so that in total, we made 100,000 comparisons with siblings. Results showed that most brothers of true contributors had a likelihood ratio supporting exclusion or having a limited support for inclusion. We have also seen that some comparisons led to very large likelihood ratios, sometimes in the order of billions. And although it may seem scary, seeing those large likelihood ratios for non-contributors should not make us panic. This is in fact a result that was expected, and Dr. Moretti explained that very good in our presentation. Because we simulated a huge number of siblings, we were expecting to have some of their brothers that would randomly share several alleles with the person of interest, and that would fit well in the mixture. But what is important to remember is that large likelihood ratios were obtained in a very small minority of the cases, with less than 1% of comparisons falling in the very strong support category. So generally speaking, the brother defense is not much of an issue when you have a very strong likelihood ratio for the person of interest. And if we ever need to give an opinion on a specific result and the genotype of the sibling is not available, the sibling LR can be used in those specific cases. So that's all I had to say. I would like to thank Dr. Moretti for putting together this webinar that has been very interesting so far. Thanks also for inviting me so I could share our research. So thanks for listening to me, and I will now stop sharing my screen.
All right. Thank you so much for that information. That was a beautiful study and a beautiful representation of how to demonstrate the results. That's very well done. Thank you. And with that, I would like to introduce Dr. Duncan Taylor and invite you, Dr. Taylor, to start sharing your screen for your presentation.
Okay, I'm just sharing my screen now. So, good morning from all the way down in South Australia here, and a special shout out to anyone of my colleagues who has got up into the early hours of Thursday morning to see this webinar in Australia. It has been very interesting, and I hope that my talk can also live up to the previous two for today. I will be talking about big data, so lots of results, and how we can look at large numbers of results to judge the performance of probabilistic genotyping systems. And in doing that, I'm also going to talk about different types of plots, or different ways that those results can be displayed, how they're related to one another, and hopefully give people a bit of an explanation for what some of these plots mean when they're reading papers that have strange-looking plots. And also I'm going to talk a little bit about multi-lab studies. So my talk is going to be rather general. It's not about any specific validation work that I've done or my lab's done, but it's going to be sort of a general overview of these sorts of things. So as we've heard today, and no doubt in the previous webinars, there's many continuous probabilistic genotyping softwares that exist that can analyze DNA profiles, and lots of those different systems have published and unpublished validations, and really the purpose for doing these validations is to test the performance of those probabilistic genotyping systems, really test their limitations to see when you can and can't use them, and ultimately this is going to lead to a judgment by your laboratory as to whether or not that system is fit for purpose for you to use on Casework. Once you've generated all the data doing these validation studies, there's a variety of ways that they can be displayed and interrogated, but ultimately you're left with hundreds or thousands or tens of thousands or even millions of numbers that you need to interrogate in some way to actually make this judgment of whether or not your system is fit for purpose. And this is usually some graphical way of showing the data so that you can see trends and make conclusions from those trends. So as I say, I'm going to be working through some of those ways of data summary and trend recognition in big data sets. So I'm going to start by just setting up a general layout, and you would have seen this, I believe, probably in previous webinars, but also today. We have on the y-axis here, so the vertical axis, an indication of the strength of support for someone being a DNA donor to a sample. We have that big horizontal line which represents neutrality, that is a log likelihood ratio of 0 or a likelihood ratio of 1. Anything below that line is support for their inclusion, with increasing support getting lower on the graph, and anything above the line is support for inclusion, with again increasing support getting further above the line. And on the horizontal axis we have, in this case, an amount of DNA. We start on the left, no DNA, and then we go to the right, and we have ample DNA. And the first thing we might think about is what would be a perfect system, and I'll call this the oracle system. So this is where you compare a number of contributors to mixtures and a number of non-contributors to mixtures of different quality, DNA amount, complexity. And an oracle system will be able to tell you unambiguously in every single instance whether someone is a contributor or a non-contributor. So you can see I've added those categories of log LR of infinity and minus infinity, which is to say it can always tell you exactly when someone's a contributor or not. So support for one proposition over the other is absolute in this oracle system. The amount of information doesn't dictate performance in any way. There's no misleading LRs or adventitious matches. And ultimately then there's no need for statistics at all. So this seems like some kind of nightmare world for me. But I don't have to worry because obviously oracle systems can't exist in reality. They're simply impossible to achieve. So instead of thinking about an oracle system, we need to think about what do we want from the best possible system that exists within the bounds of reality. Now, typically that would be something like we know that if a system has certain amounts of information that can be used to discriminate between possibilities, as you get less information, you have less ability to discriminate, and if you get more information, you have more information to discriminate. So we couple this with the fact that the lab hardware and DNA profiling kits are very sensitive and can produce complex mixtures, and many of these are beyond our ability to interpret. So this is why we've produced and developed and implemented probabilistic genotyping systems. So we need some way to carry out a valid evaluation of how they work. And once you do that, as well as having all this data, you're left with a question of how do I validate that this system is giving me the right answer when the answers are by definition too complex for me to know if they're right. Okay, so that's looking at those answers in a very close-up level. So that's looking at individual answers one at a time. But what you can do is take a step back and look at the data on a much larger scale, and you can start to, instead of thinking, is this specific number right, you can start thinking about, is this data set showing the trends that I would expect from a probabilistic genotyping system that's working properly? And that's what a lot of these validations that are done really do. They set up a number of mixtures, different complexity, different amounts of DNA, and they start looking at trends in the likelihood ratio for either support for inclusion or exclusion, contributors or non-contributors, and they make judgments on whether or not the probabilistic genotyping system is working, given what trends are being seen. So the overarching general theory that you use when you're predicting these trends is that as a model is provided more relevant and correct information, its ability to distinguish from true and false hypotheses should improve. So when you're talking about information, correct and relevant information, in the case of evaluating DNA profiles, you're talking about information that's in the electropherogram itself, such as peaks, the allelic designation, peak heights, molecular weights, and there's also other information that can be provided which is more case-based, case circumstance-based, such as whether there's any people that can be assumed as a contributor to that mixture. You might be able to provide some information about relatives being present in the mixture as well. And all of this information, in its ability to distinguish these different true or false propositions, really comes down to the power that that information has to inform us of the genotypes of the contributors. So at the ultimate end, if you have enough information about a particular contributor, you'll be able to resolve its genotype completely and absolutely across the entire profile, and then you get a likelihood ratio that's very close to its single source likelihood ratio. All right, so what we're going to do is use this basic overarching theory of information distinguishing true and false hypotheses and build up some predictions of how we expect a probabilistic genotyping system to work in an ideal setting. So here we have our architecture for how we're going to show these results: DNA on the x-axis, support on the y-axis, and we're going to compare a number of known contributors and calculate likelihood ratios, which will indicate in blue, and compare a number of known non-contributors, calculate likelihood ratios, and represent them in red. And if we did that for DNA profiles where the contributors all contributed ample amounts of DNA, we would expect quite high levels of discrimination. That is, we would expect the contributors to give high inclusionary LRs and the non-contributors to give strong exclusionary LRs. And we might ask ourselves, what happens if we increase the complexity of the mixture? And both our previous two speakers spoke on this topic, so I won't spend too much time here. But as you increase the number of contributors to your DNA profile, the model loses power to resolve any single contributor's genotype. This is basically a form of loss of information, loss of ability to distinguish true from false hypotheses, and so you would expect with that increasing complexity, your likelihood ratios would contract towards 1, or in this case, a log likelihood ratio of 0. So you would hope that your contributors still gave LRs that supported their inclusion, but you would expect them to be weaker in their support than when you had very simple profiles. And similarly, you would expect non-contributors to still give likelihood ratios that supported their exclusion, but again, that level of support would be weaker than when you had very simple DNA profiles. And we can consider again what would happen if we changed something else. So this time, instead of changing mixture complexity, we'd change the amount of DNA that goes into the mixture. And again, we have this idea of losing information, because as the amount of DNA decreases, peak heights become lower and more variable. They are less able to inform on the template that gave rise to them. Therefore, again, a lower ability to resolve on any particular set of explanatory genotypes, and likelihood ratios contracting towards 1. So we can see we have this sort of triangular effect here. With very little DNA, all the likelihood ratios will start at 1, or log likelihood ratio of 0. Intuitively, this makes sense because you could think of the ultimate endpoint here. If you had a DNA profile that was completely flatlined with no information, by definition, it's given you no information to distinguish a contributor from a non-contributor, and all likelihood ratios are going to be 1. And as you get more DNA, your ability to distinguish true and false propositions will improve. Finally, what we would expect if we add more correct and relevant information is an increased ability to distinguish true and false propositions, and we would see this in our likelihood ratios by increasing support for inclusion of known contributors, increasing support for the exclusion of non-contributors. So an expansion of the likelihood ratios away from 1. So just from theory, without doing any actual experimentation, we can build up a series of expectations so that when we then go into our validation work, we can check it for adherence to these predicted trends, and then that's going to assist us in determining whether or not we think this probabilistic genotyping system is fit for purpose and is working properly. So here's an example of exactly this sort of experiment being done, and what we see in reality. Obviously, reality is a little bit more stochastic than an ideal world, so the lines here are a little bit wobblier, but you can see we've got our log likelihood ratio on the y-axis. We've got an amount of DNA on the x-axis there, starting from very, very tiny, one picogram on the left, going right up to 500 picograms on the right. And we've got likelihood ratios for contributors and non-contributors in green and red, respectively, for 2, 3, and 4 person mixtures. What we can see is that as you increase the complexity of the mixture from 2 to 3 to 4 contributors, you get that likelihood ratio trend line contracting towards 1. And as you decrease the amount of DNA, then you see the likelihood ratio contracting towards 1. So this is following the trend that we expect. We would use this information to judge that probabilistic genotyping system was performing properly. As an example of adding information, we could do multiple PCRs, or we could assume some of the contributors and just compare the remaining contributors to those profiles. And here we have an example of a graph that does exactly that, where the original mixtures, these are all 4-person mixtures, are shown in thick lines, green and red, which are the closest to the x-axis. If you add multiple PCRs, those LRs expand away from 1, and if you then assume people, the LRs expand away from 1 again, indicating that you're getting that increased information that you're giving it is increasing its ability to distinguish true from false propositions. So again, we could use this information to determine that our probabilistic genotyping system was working properly. Another piece of theory that is important is not just the absolute power of the likelihood ratio, but also that it's properly calibrated. And there's a really good paper here by Ramos et al. Talking about calibration of likelihood ratios that sort of boils down to invalidation studies. This idea of the adventitious matching, so a likelihood ratio of X will be wrong 1 over X of the time. Or in other words, if you had a likelihood ratio, for example, of 1 million, you would expect 1 in a million randomly chosen non-contributors in the population to give that sort of level of support. And another way of putting that, which has been put in the past in literature, is the likelihood ratio of the likelihood ratio is the likelihood ratio. That should work. So we've got an example here of a well-calibrated system on the left. I'll see if I can bring up a laser pointer. Here's a well-calibrated system of likelihood ratio calculation on the left. We can see it's relatively clear that the true contributors are giving likelihood ratios that support inclusion, and the non-contributors are giving likelihood ratios that support exclusion. Whereas over here we have the exact same two distributions shown, but it's poorly calibrated because we can see both the contributors and non-contributors are giving inclusionary likelihood ratios. Even though there's a difference between those two distributions, it's a poorly calibrated system. And you can see this if you do specificity tests. So you carry out a whole bunch of calculations of likelihood ratios of non-contributors, and again you can predict what you would expect to see in theory and compare this to what you actually see in practice. But in theory, if you had a single-sourced DNA profile up the top here, and you compared it to a whole number of non-contributors, you'd expect a lot of likelihood ratios of 0, which is to say that they're excluded. And if in this case the single source or the random match probability was 1 in a million, you would occasionally see a little adventitious link giving a likelihood ratio of 1 million for a non-contributor. If your mixture complexity increased a little bit, so you had some medium complexity, not all your likelihood ratios would be exclusionary now. You'd get a number of them which gave mild exclusionary support, and some that gave mild inclusionary. And then if you had a very weak profile, then you would expect a distribution of likelihood ratios that were relatively neutral and around 1. And what you could do is also compare that to your distributions that you would get if you had known contributors that you were comparing to the mixture. And it's almost the mirror of what you see for non-contributors. So here your known contributor compared to your single source profile would give you all inclusionary support at the value of 1 million. For the medium complexity mixture, you get lower levels of inclusionary support for known contributors. And for your very weak mixture, you would get relatively neutral support again, because you have very little information to distinguish contributors from non-contributors. We can do that in reality. And what I've got here is the results. In this particular case, we've taken a 2-person mixture in a 10-to-1 ratio. And we've generated a distribution of likelihood ratios for non-contributors and a distribution of likelihood ratios for contributors. Now, in this particular case, because we're talking about a single mixture here, generate these non-contributor likelihood ratios by sampling genotypes at random from the population and comparing them to the profile. And we generate these distributions of contributors by taking samples of genotypes from the results of the deconvolution. So genotypes that are not excluded, that have some weight coming out of the deconvolution. And so this represents your H1 true or HP true distribution. And this is what we see in reality. This is sort of an all-in-one proof of concept of our previous in-theory distributions that we were expecting. So in this 2-person mixture, we have major, where we would expect quite high levels of inclusionary support. We have this lump of blue here, which represents the LRs associated with the major. And we can see the log likelihood ratio is quite high here. It's close to 30 orders of magnitude, representing very strong support for their inclusion to that mixture. And then we have the minor contributor here, still giving mainly inclusionary support for true contributors, but much weaker than the major. So we're seeing this expectation of, from theory that we were looking at in the previous slide, playing out in reality when we do it in practice. Again, marrying up our predicted trends to our observed trends gives us an indication that the probabilistic genotyping system is working well. So going a little bit further beyond this, predicting these trends, a common problem, not just in forensic science, but across a number of sciences these days, is that we have huge volumes of data. We need to decide what we're going to do with it. And half the battle sometimes is just figuring out the best way to summarize it that makes sense, that's going to show you the particular trend that you're interested in showing, and allow some predictions to be made and shown in this way of displaying the data. There's an enormous number of ways that data can be displayed, and I'm just going to go through a few of the most common, how they're related to each other. So all of these plots that I show you in the next block of my talk here, all are looking at the idea of comparing individuals to mixtures and providing yielding likelihood ratios at the end of that for contributors and non-contributors. And we've seen these sorts of plots numerous times. They're scatter plots, or often called sensitivity or specificity plots. Y-axis relates to the strength of support for inclusion or exclusion. So we have this log likelihood ratio of 0 here, and this line across the middle, which represents neutrality. The x-axis relates to some dependent variable. So in this case, it is the intensity of the peak heights, or the DNA amount that contributor that went into the mixture. And we spread the data across the x-axis so that it allows us to see the level of how that level of support trends with respect to that variable. So how does the level of support in this case trend with respect to the average peak height of that contributor? And what we can see is the expected trend of contributors giving increasing support for their inclusion to a mixture as peak height increases, and non-contributors giving increasing support for their exclusion to the mixture as peak heights increase. And these can be broken up into their individual components of comparisons to contributors and comparisons to non-contributors in order to better show their individual trends we desire. So the pros and cons of this type of plots, well, the pros are that the data is not summarized, so there's no loss of information. You can, in theory, see any particular point in the analysis that you would like. Also, you can see trends, as I've described, or as I've shown the arrows there, quite easily for a number of different aspects of data that you might look at. It doesn't require any binning across the intensity categories because it's treated as a continuous variable in this particular graph type. And quite often these are used because they have an intuitive interpretation. So you don't need much specialized knowledge about how the graph type works and how to interpret it in order to understand what you're being shown. The cons of using this type of display of data is that when you get a lot of data, you can reach a bit of visual overstrike, so you have so much data displayed here, it can start to obscure the trends. And you find that scatter plots, or these sensitivity specificity plots, don't necessarily provide a good feel for the distribution of the likelihood ratios at any particular peak height or DNA amount, because there's so much data overlapping. So what you can do is, instead of showing the likelihood ratios in a scatter plot here on a continuous variable for a peak height, you can break those peak heights up into categories. And we see here on the right, we've got a category of peak height from 0 to 100, 100 to 200, that should be 200 to 500, and greater than 500. And so what I'm going to do is now take a slice of the HD results for the first category from 0 to 100. I'm going to translate that to a distribution onto my graph. See here now, we're still on the same scale of log likelihood ratios, but we can see that this data here that is shown in a scatter plot is now instead shown as a distribution here, with the width of this plot representing the number of points at that particular region. So 0 has the greatest number of points in the 0 to 100 category for non-contributors, and then we can see that gets less and less. So we have minimum value here of about minus 15 or so, and the maximum value of probably about 6 or 7. And in that same category, we can also take a slice, or the same slice, for the comparison to known contributors. And we draw that plot, and we can see here again, the width of the plot represents the number of points at that particular log likelihood ratio value. And we can do that for the various other points or brackets of intensity that builds up what's called a violin plot. And this is now a nice way of showing the distribution of data in different intensity brackets. So the y-axis still relates to the strength of the support for inclusion or exclusion. The H1 or HP or H2 or HD are shown by different colours on this particular plot. So here we have the HP and the lighter colour is the HD, and the width of the shape designates the number of points at that level of support. And it's usually smoothed by some smoothing algorithm, such as a kernel density function. The other plot that is very similar to a violin plot, which you might see in a number of different papers, is called a box plot or a box and whisker plot. Whereas the violin plot provides a smooth visual indication of the density of observations across the range. The box plot will tend to show that just interquartile ranges rather than the density. So the general setup of a box plot is that you have a median value which represents that central line within the box. You have quartiles which represent a quarter of the data in each quartile. The width of this quartile 1 to quartile 3 range is known as the interquartile range, and then you generally have some little what are called whiskers at the end here, which are either the quartile 3 plus 1.5 lots of the interquartile range, or quartile 1 minus 1.5 lots of the interquartile range, and then anything beyond that is considered an outlier. So in this particular violin plot, we would end up with something like this here. So we can see these points represent the median of these plots, and then you have the interquartile range within the box, and then the whiskers to show either the maximum range or this plus or minus 1.5, the interquartile range. So the pros of this type of plot are that it more clearly shows the distribution of values, that's not obscured by overlapping points in a scatter plot. It's a nice clear way to visually summarize a lot of data, and this violin plot also shows the density at any particular value, so that shows a bit more information. These types of plots are particularly good. Use also when you have a variable that's categorical, so rather than being continuous like DNA amount or peak height, you might have categories such as 2-person, 3-person, 4-person, or likelihood ratio from star mix, likelihood ratio from true allele, likelihood ratio from LR mix as different categories. They're often displayed then in box plots or violin plots. The cons of these types of plots is that if you are using it to display a continuous variable such as DNA amount, the data needs to be binned into sort of arbitrary categories. And also, to some extent, although they're generally understandable, but to some extent, you need to know how they're constructed in order to really interpret them properly. So, for example, if you're looking at a box plot, you sort of get even if you don't know how they work, you get a general feel for the spread of the data, but to really interpret it properly, you need to know what this line represents, the median, and what an interquartile range is and how they're constructed. Okay, so the next type of plot that you'll sometimes see is a density distribution. So this is where we sweep across all of the points in a particular category. So in this case, we'll consider the HD category, the non-contributor category, and we're going to sweep across from left to right, and we're going to tally up the density. I did the HP one first. So we're sweeping across the HP plots, and what we're going to do is tally up the number of points at any particular value. So you can see here, if I just go back, as we sweep up from the bottom, we sweep up to the top, what we're going to see is the line slowly building up on the right-hand side here. So its maximum is sort of in this middle zone here, and that represents this middle zone here. And we can sweep also across the HD true results. We would get a density distribution here. So these are quite similar to violin plots in that they show a distribution of values across the range of likelihood ratios you can get. And an example in literature is from a paper by Kelly et al. And here we can just see the distribution of HD true LRs when you consider mixtures 2 or 3 people. And there's lots of different, I've just picked out one random example of a density plot, but there's lots of different examples in the literature of this type of thing. Now, I said that violin plots and density plots were very similar, so you can actually think of them as being virtually the same thing. What I'm going to do is here draw a bunch of lines for the HP true and the HD true LRs onto the violin plot, and the violin plot is center aligned, so these arrows are center aligned. And if you push them all to the left, so they're actually aligned, the right hand side, now they're all nicely right aligned. And what we're going to do is rotate the graph, so 90 degrees, and then draw new lines that follow the top of these arrows for each of the 2 different distributions. Then in fact what we end up with is our density plot. The way that you can convert a density plot to a violin plot is basically you're going from a density plot to a violin, you'll center align it and make it vertical. You're going to go the other way, make it horizontal and right to justify it. But other than that, they're showing the same data. So they have similar pros and cons. So a density plot is a common way to display data, and it's generally quite intuitive to get the gist of what's going on. When you look at these plots, it's easy to see overlap between the distribution at critical points, such as the point of neutrality for a likelihood ratio, and it's easy to see when the method is not calibrated well. The cons, so as with a box or violin plot, it may require binning of data if you're interested in a particular trend across a continuous dependent variable, and it can sometimes be a little bit difficult to understand the meaning of the y-axis, in that it's a density, it's not a probability. That value of a density comes about because the area under these graphs need to equal 1, and so that density can be any number from 0 to greater than 1. But it's not necessarily intuitive to people what a density is. So what you can do is convert that density to a probability, and that converts your density distribution to a Tippett plot. So if we draw the HP true density distribution, but you'll see on the right hand side, instead of going back down, it kicks up to the top here. That's because this graph on the right hand side is showing the cumulative value, as in the area as we go along line. I said that before the area is equal to 1, so all these Tippett plots, or these cumulative distribution plots, will go from 0 to 1. And we can see that for the HP true, and we can also see it for the HD true. These are a little bit less common in forensic biology, but they are quite common in other forensic disciplines, so you see it often in voice speaker recognition and other things like more comparative sciences such as that. But here is a couple of papers that do talk about them specifically for the DNA. The pros are that the y-axis is now probability, so it goes from 0 to 1, and so it's intuitive to interpret. You can sort of look at this particular plot and you can see at our interesting point here of LR equals 1, so our line of neutrality, we can see the HD plot here crosses it at about 0.95, so there's a 5% probability of getting a misleading LR. You can read them in that way, and the same for your HPs, but the other way around. Again, it's easy to see calibration of the data, and the different areas of those plots have have intuitive meanings that are relevant in DNA profiling analysis. The cons, I guess, again, binning may be necessary, and the other con is that it's a summary, and so it doesn't show any individual values, such as the scatter plot. All right, the final transition between plots, or the final relationship I want to show, is the difference between how you get from, say, a tippet plot to a ROC plot. A ROC plot is a receiver-operator characteristic plot, and it shows, instead of the likelihood ratio and probabilities or densities or DNA amounts or anything like that, it shows the false positive rate compared to the true positive rate. So, as an example, we'll pick a threshold of log LR of minus 40, and we'll consider, if we use that threshold of log LR of minus 40, how many true positives would we identify? So, in other words, how many of these HP true values are above a log LR of minus 40, and in this case, all of them are. So our true positive rate on this axis is 1. And we ask the same thing, how many negative results, how many true negatives, so this HD true line, if we use the cutoff of log LR of minus 40, how many negatives would we identify? And again, we would identify all of them. So our false positive rate would also be 1. So we'd end up with a point here on a ROC plot. We could then move across a bit, and we'd use a threshold of minus 20. We can see our true positive rate is still 100%, because still 100% of the HP true data is to the right of that particular cutoff, but not all of our HD true LRs are. So we move in slightly on the false positive rate axis, but we're still at 1 on the true positive rate axis. And we can continue to move with this sliding threshold of likelihood ratios all the way up the graph until we would end up with a plot that looked like this. Okay, so this is translating tippet plot in this particular example to its equivalent ROC plot by looking at true and false positive rates. ROC plots are generally used to make decisions. So, for example, if we wish the outcome of our comparison of a reference to an evidence profile to be that we said that someone's DNA present was not unequivocally, which I don't advocate, but if we wanted to make that decision as to whether someone's DNA was there or not, and we considered both the true positive rate and the false positive rate as equally desirable outcomes, then the optimal decision point is the point of the threshold on this particular curve that is the furthest distance away from that diagonal line. So this is the classic use of a ROC plot to make a decision. And the other classic use is that you can calculate the area under this curve and use it as a measure of performance across a data set. So, for example, you could compare the number of donors and non-donors of DNA to a number of different mixtures, one particular probabilistic genotyping system, and produce a ROC plot and calculate this area under the curve. You could compare the same individuals to the same mixtures in a different probabilistic genotyping system, produce this ROC plot, calculate the area under the curve of that second ROC plot, and then your comparison of the performance of those two different probabilistic genotyping systems could be measured by the comparison of the area under the curve, noting that what you're doing is contracting the entire performance of all of the data down to a single number for comparison, which has its inherent dangers. Okay, so here's some examples of ROC plots in reality. So for example, here we have some 5-person mixtures, and these are pulled apart into bins of intensity, peak intensity, so 0 to 200 RFUs, 200 to 500, 500 plus, and the rock plots, which go pretty close to the top left-hand corner. Here you can see the axes are called FS and CS, so in this particular instance it's false support and correct support, but it's exactly the same thing as false positive rate and true positive rate. And what you often see is rock plots displayed with the axes modified in some way, so either it will be a close-up of this corner, or it will be displayed on a log axis, because really all the interesting part of the rock plot is up in the corner there. So here's an example in the literature where it's been used exactly the way I described to compare different probabilistic genotyping sets on the same data. Different probabilistic genotyping systems on the same data sets. Okay, so the pros for rock plot, they're good where you want to make a decision, and they can be useful for comparison performance in two different methods on the same data set. The cons are when you're calculating an LR in an evaluative sense, you're not generally looking to make a decision. You're not looking to decide whether someone's DNA is present or not, and it doesn't indicate whether or not the method is poorly or well calibrated. In fact, if your two distributions are the same but poorly calibrated and well calibrated, you'll get exactly the same rock plot. Also, the con is that they're not as intuitive to understand their meaning unless you really know what they are, and you lose the sense of an absolute magnitude of the LRs that you're getting. Alright, so here's sort of my overall summary of all the plots and how they're related, just in a quick one slide, sort of take-home slide. With your scatter plots here, you summarize them as a distribution, you get density plots. You vertically center align those plots and you get violins. If you then go from densities to quartiles, you get box plots. You change your density to a cumulative distribution, you get a tippet plot. And if you display it, a plot by false or true positive rates, you get a rock plot. So this is sort of how they're all interrelated to one another. Okay, so that's sort of the first half of my talk. The second half, I want to talk about some interlab studies. And these are really good for a number of reasons. Most obviously, you can amass large amounts of data that would otherwise be too costly for any individual lab to do so, and that gives you greater ability to look for these trends you predict should exist. And what you can see is how profiles are evaluated. If the study spans different evaluation systems, you can see how a particular system is being used. If that's the interlab study is designed to concentrate on a particular evaluation system, you can have a look to see how profiles are being interpreted prior to evaluation, or you can look at how different lab processes affect the profile interpretation variability. There's a whole bunch of different types of interlab study and set up, and I've sort of tried to pick a few different examples of different types that I'll quickly go through here. So probably one of the most well-known amongst the audience here, I'd assume, would be the NIST studies, particularly mix 5 and mix 13, where a number of different mixtures were sent out to labs, 4 and 5 cases for mix 5 and mix 13, and the mixtures ranged from simple to very complex and very challenging. There was 70 labs that participated, or 69 labs that participated in mix 5 and 108 that participated in mix 13. So this is what I would call a state of play or a state of the nation interlab study, in that it wasn't necessarily trying to control for any particular variables such as probabilistic genotyping or interpretation method. It was simply putting the mixtures out there to see how much variability there was in the world. And as you can imagine, in this sort of study, there was quite a lot. So because the different labs that were involved, they spanned the hardware, the profiling kits, the thresholds, the evaluation systems, and the interpretation guidelines used to evaluate the profiles, there was an enormous range of results that were obtained. So these are 3 examples of box plots broken up by their category of whether CPI was used, whether the LR was used, or the random match probability as the weight. And these should all be inclusionary, and what we can see is that they almost all three examples of these mixtures have inclusionary values that range from about likelihood ratio of 1 to about the ratio of 10 to the 20. So really, this is an enormous amount of variability, which I guess was not surprising, but people probably hadn't been shown it in quite such a summarized, confronting way as this beforehand. So most controversial of those mixtures was the mixed 13-5 case, case number 5, which was a mixture of 4 people, but with genotypes chosen that no more than 4 alleles occurred at any one locus, and 3 references were supplied. 2 of them were donors and 1 non-donor, who was C, but this non-donor C had a lot of allelic overlap with the other donors. So this one is particularly controversial because while C should be excluded, the study found that 74 of the 108 labs included reference C in the mixture. 23 found the mixture too complex to interpret. 4 found the mixture okay, but C was inconclusive. 7 excluded him, and not part of the study, but within John Butler's paper here, NIST Interlaboratory Studies, there's a mention to say that true allele star mix DNA viewer mixtures all excluded C from the mixture, which sort of shows the power of these probabilistic genotyping systems, utilize all those little micro bits of information in peak height, and gives you really good discrimination power between contributors and non-contributors, even when there's a lot of allelic overlap. And there's a bit of follow-up work done by Buckleton et al. That looked in particular at mix 13 and concentrated a bit on mixture 5 and showed how different systems performed on mixture 5. Okay, here's another example of a interlab study. This one was from Benshop et al. And there were 26 participants in 3 labs looking at 5 NGM profiles. And they were asked to determine whether or not the profile met the criteria for comparison to a reference, and then the results of the comparison, and whether or not a statistical weighting would be calculated, and if it was calculated, what method was used. Again, this is a bit of a state of the nation paper, and there is a number of different methods used. And as you can expect, there's a fair bit of variability, which was largely related to the complexity of the profiles. So the graph on the left here just shows all the different 5 different tasks they were asked to do, each with different sets of profiles. They were asked to determine the number of contributors to those profiles. In some cases, that was very easy, and they all determined the same number of contributors. In other cases, it was a lot more difficult. So, for example, task 2, set B here, there was profiles across the 3 labs that were interpreted as either a 3 or a 4 or a 5 or a 6-person mixture. And once again, reflected in that was whether or not statistical weighting could be carried out or whether or not they would interpret it. So the different labs here indicate whether it was inclusionary and they would give a weight of evidence, whether it was exclusionary or whether it was uninterpretable. And you can see different labs performed or gave different results. Okay, this is an interlab study which concentrates on a particular probabilistic genotyping software, in this case StarMix. So this is controlling for that particular variable, and the aim of this particular study was to see whether or not there was standardization in amongst the labs that had implemented the same probabilistic genotyping software. 20 people were involved, about half of those from one lab and the other half scattered across other labs in Australia. 3 samples were sent out that were identifiable profiles, and each of the labs were asked to interpret the profiles and use StarMix to calculate a statistical weighting. Now, the biggest difference here came down to the choice of number of contributors. For case 1, everyone chose the same number of contributors and got a very close likelihood ratio to one another. In the second case, you had some people that called it a 2-person mixture, some people that called it 3, and some people that called it a 4. And those that called it a 2-person mixture, their likelihood ratios clustered, and those that called it a 3 or a 4, they sort of clustered with each other, but were somewhat separated from the 2-person likelihood ratios. And also in case 3, there was some discrepancy between whether it was 3 or 4 person mixture, but those likelihood ratios generally sort of clustered around one another. So the conclusion of the study really was the differences mainly came down to assigning different numbers of contributors. There was some differences caused by different propositions being set up, but it was mainly number of contributors, and it wasn't necessarily to do with interpretation rules, as half of the 20 people were from the same lab and had assigned different numbers of contributors within that lab. Okay, here's a interlab study. Again, this is slightly different. So rather than sending mixtures out to a number of different labs and asking them to interpret the mixture, in this particular study, or interlab study, a whole bunch of data from different labs was all brought together and compiled as one big set. So again, that's a different style of doing an interlab study. And this was carried out in particular to look at the performance of star mix with regards to a number of aspects that had been brought up in the PCAST report. That's for the President's Council of Advisors on Science and Technology, which was questioning the ability of probabilistic genotyping systems to interpret or evaluate profiles greater than 3 people, and where the person of interest was more than about 10 or 20% of less than 10 or 20% of the profile. And in that PCAST report, they identified a number of areas that they would like to see further work or exploration into. So that's what this study was about. So it particularly concentrated on the number of contributors, the amount of allele sharing, and the amount of DNA of those contributors in all those different mixtures. It was a large compilation from many different labs. So you can see here the numbers of mixtures involved was quite large, and it spanned different lab hardwares, profiling kits, and analytical thresholds in the data that was received. It was summarized in a number of ways. So this is a good example of, if you look through this paper, you will see a whole bunch of the different types of plots that I've talked about in the first half of my talk. So we have some violin plots here, breaking up the data into different peak height categories and showing the ability to distinguish contributors from non-contributors. To summarize it all in a very quick way, the data sets generally showed good discrimination power. Another interesting point of the study was looking at the number of contributors that were assigned to the mixtures and comparing that to the target number of contributors. So here we have the concept of target number of contributors compared to assigned number of contributors. So just very briefly, a target number of contributors is the number of contributors that a mixture is made up to be. However, you have this sort of philosophical idea that if you generate a mixture of, say, 4 people with one person who's there in very tiny trace amounts, and then you dilute that down to the point where that very tiny person hasn't donated or contributed in any way to any of the fluorescence seen in the profile, then really they shouldn't be considered a contributor, because by definition, a contributor to a DNA profile is someone that's contributed some fluorescence. So then you have the correct number of contributors. So that's how many people have contributed fluorescence to what you see. And then you have the assigned number of contributors. So now this brings in the idea of using an interpretation threshold to assign an actual number. But interestingly, what we can see for 3-person mixtures, almost all of the mixtures were called 3-person mixtures with some overestimation. 4-person mixtures, the majority were still assigned as 4, with about a quarter of the time of being underestimated and a very small amount of overestimation. And as you went up to 5 and 6, you increased the number of profiles where underestimation occurred. So when it got to profiles that were 6 contributors, or the target number was 6 contributors, the assigned number was always less than 6. And the other interesting thing about this study was that it looked at the amount, or the way that allele sharing affected the ability to discriminate contributors from non-contributors. And in fact, that one of the findings was once you adjust for the number of contributors to the mixture, then actually the amount of allele sharing when using a probabilistic genotyping system didn't have a huge effect on that discrimination power. So that was good. That was showing the power that peak height has to inform us of the genotypes of the contributors beyond just simply the presence or absence of peaks, even in quite complex mixtures. Okay, another interlab study, which is a quite recent one, again looking at star mix 174 participants and using a couple of complex profiles from the approved data set. This one found here one example of some variability seen in the likelihood ratio of one of the known contributors. And just to very briefly summarize, the greatest sources of variation in the likelihood ratio in order, again, were assigning number of contributors, followed by exclusion of some of the loci by some labs, followed by difference in the capillary electrophoresis settings. So that was the settings within, I think a lot of these labs were using GeneMapper. And then lastly, the MCMC run-to-run variability was the smallest source of variability shown. We have here a GHEP ISFG collaborative study on some mixtures, and again, so this is a bit of a state of the nation because they weren't controlling for any particular probabilistic genotyping or interpretation method, but it happened to be that most labs were using LR-Mix Studio. They had 25 participants and 2 NGM samples, and we can see quite a large range in this particular case of LRs, so ranging from 10 to the 3 up to 10 to the 15. And largely these differences came from different software settings and the way that people were treating stutters or applying different thresholds to remove or keep on different peaks, and the choice of the different loci ignored. So this one largely came down to, rather than assigning a number of contributors, it came down to thresholds to do with the interpretation schemes in the different labs. Here is an LR-Mix interlab study, looking at 2 profiles across 18 labs. This study achieved a very high level of reproducibility or consistency between the labs, and that was put down to using the common software tool, standardizing the protocol for analyzing these profiles, and also standardized training for those different labs. So in this particular instance, those 2 profiles were analyzed in the same way, labs. And I thought I'd just put this one in, not because it is anything really to do with probabilistic genotyping, but I thought I really like this paper by Steensma et al. Because it takes it right back to the exhibit level. So here's an interlab study where they sent out a number of cable ties where people had handled the cable ties and zip tied them around some pencils. They sent those cable ties out to the labs, got the labs to sample them and extract them, look at the profiles, and then evaluate the profiles or interpret them with respect to whether or not someone is a contributor, and then apply that finding to, in this case, an activity level evaluation. So it was really the amount of variability you see in the entire process, and it sort of puts the probabilistic genotyping interlab variability into a wider context. And quite pleasingly, they found that even though the profiling results were quite different, they found that the effect on the activity level evaluation wasn't so huge, but I certainly recommend reading that paper. It's quite a good one. And the very last thing I want to mention is a paper which you may have already been talked about in the webinar series, which is a Ladio et al. Paper, which wasn't an interlab study. It was all within the one lab, but the one lab looked at the same profiles on different probabilistic genotyping systems. So it sort of mimics like an interlab study, but instead of controlling for a particular probabilistic genotyping system amongst multiple labs, you're controlling it within one lab, so you're standardizing things like profile generation, interpretation, training, and that sort of thing, applying that controlled interpretation scheme to different probabilistic genotyping systems. And again, very pleasingly, in this study, they found that all the probabilistic genotyping systems, or the continuous ones, are tending to converge upon the same sort of likelihood ratios for the same mixtures, which is what you would hope is happening. All right. What do they all show? So there's many points along the route where variability can be introduced. So from the exhibit, how it's sampled, extraction, PCR, capillary electrophoresis, assigning number of contributors, interpretation, deconvolution, reference comparison, the method of statistical weighting, and then how it's reported. Some of these can be standardized with education. So, for example, the interpretation, assigning number of contributors, reporting. Some of them can be standardized with software, so the deconvolution and reference comparison part, and some of them can be standardized with alignment of hardware or lab wetware, so all the lab processes basically. But all of them have some level of inherent and unavoidable variability within them. So what we take away from this are these interlab studies what they want? It sort of depends on the question a bit. Possibly not for validating probabilistic genotyping systems. At least they have sort of limited advantage over what can be done in a single lab, but they're really good as a means to assess interlab variation at a macro level and potentially identify areas for improvement in the interpretation scheme, methods for deconvolution, proposition setting, and those sorts of things. I just want to show this study, which you may have seen before, but I really like this paper by Bright et al., which showed the amount of variability you see in the likelihood ratio from the MCMC process, but also from different lab processes. So Bright et al. Looked at the amount of LR variability when you take different samplings of a population database, and within each of these different 10 samplings, the points that are vertically aligned are the MCMC variability. You can see the MCMC variability is quite small compared to the larger population database variability. Here we have injection number on the C instrument, so which if you're using the same capillary to inject the sample multiple times, what effect that can have on the likelihood ratio. Again, you can see it's quite large, so from 80,000 up to 1.3 million, whereas the MCMC run-to-run variability is small within that. Here you had your CE load, so instead of this is the same sample injected with different capillaries on the instrument, a large CE load variability on the LR, and then you have amplification here, where you see probably the largest variability, so that LR goes from 4 up to 100 million based on these different amplifications of the same sample. So what you can do is take that data, so let's take the amplification that led to the highest likelihood ratios, and look at the HP true and the HT true distributions, and that's what's shown here. So we have likelihood ratios for this particular deconvolution, which sort of span from about 1 to about 17, and almost all of the H2 true or HD trues are excluded, and what we see here is a very small blip, which you can barely see here, which represents 0.33% of the data. And we can do the same thing and look at the distributions of HD true and HP true LRs for the worst performing amplification. We can see that the HP true is very much more down towards sort of neutrality. It sort of completely peters out by 9, whereas the other one was only halfway through. And now the HD true data, 41% is not excluded, and you can see a much more prominent amount of HD true results. And just to show the benefits of replication, what we could do is take the lowest performing amplification and add it to the second lowest performing amplification, so in this case 2. Here's our LR from the single worst performing amplification. Here is a deconvolution which includes 2 amplifications, so in this case the worst 2 performing, the LR it gives, and then the next adding in the next most worst performing, and again the next most worst, and so on and so forth, right up until you get to the point where you've added in 10 and you're adding 10 replicates all into the single deconvolution. And you can do the same thing starting at the best performing amplification and adding the second best and the third best and fourth best and so on. So what this shows is the power of amplification. If you don't have to split up your DNA extracts, there's a great power in additional information given by added amplifications. And really, the other thing that shows me is even if you have 2 pretty crappy amplifications, by the time you get 2 amplifications, you've got a large amount of power. Already it's jumping up to the performance of the best amplification when you've got 2 really poor amplifications. Final couple of things I want to show is you can look at HP and HT true distributions for all of these amplifications all at once. So you can imagine deconvoluting just this one and looking at the HP and HD true, and then deconvoluting just this one, looking at the HP and HD true distributions, and adding up these 10 different deconvolutions distributions, and you get something that looks like this. The HP ranges from about -1 to 17, and you've got this little amount of H2 true. Other way you can do it is to add all 10 of them together into a single deconvolution and look at the HP and HD true distributions, and what you can see is there's virtually no HD true results that are not excluded at all, and the HP true is much further. It shows much stronger support for inclusion. Really what we're showing here is when you take multiple measurements of a sample, PCRs in this case, it's their combined distribution that gives the analysis power. And if you treat them separately and combine them post-deconvolution, it squanders some of the power that repeated measurement is going to provide in the shape posterior distributions of parameters within that model. I realize I'm almost out of time here, but that's okay because I'm just about finished. My take-home slide. Tools have been developed that allow us to interpret profiles that are too complex to reliably interpret manually. We need to investigate the performance and limitations of these systems. You can do that by predicting trends in the data and showing that your validation adheres to those trends, and that can be done when individual data points have complex underlying mechanics that are perhaps beyond many of us to calculate manually. There's many ways the data can be displayed, and the choice of which data type will depend on the point that you're making or the trend that you're trying to see. Inter-lab studies can be useful to show how far the implementation of these systems has driven standardization and objectivity within the community. They have limited meaning with regards to the validation and the function of a particular genotyping system. That's not really their point. Their point is more a greater state of the nation kind of view of the world. Okay, and that's it for me. Thank you very much for your time listening.
Duncan, thank you so much. I really want to tell you how much we appreciate you walking us through all those papers and helping us understand the presentation of the data and make some recommendations to us as well for our own validation studies. So thanks so much for being a part of our session today. We had just one question. Thank you. Just one question that we had today. So I'm going to share my screen. So here we go, so that you can see it. There, sorry. The question for Sarah. I'm not sure if it was about your study or about just validations in general, but do you run all samples in duplicate? You could answer that about PCR or ProbGen, I suppose, and if not, what is the determining factor?
Okay, so I will answer regarding how we use replicates for casework. So in casework, we mostly do replicates to resolve some ambiguities that we might have relating to our manual interpretation of the results before using star mix. So basically, we do replicates for all complex mixtures, for example, for all 4-person mixtures, or when we have a low-level contributor that is present.
Fantastic. Okay. And that, I think that answers the question. I hope it does. And that was the only question we had today. So I think that's an indication that you guys did a great job explaining all of your material. So thank you once again. With that, I would like to try to flip back over just to our schedule. I can find that in my talk here, just as a reminder that, where are we? Just as a reminder that next week we are, sorry, I'm failing at my little exercise here. Next week we are going to be off, and then on June 12th, we'll be returning to talk about the representation of statistical weights to stakeholders in the court. So the same time in 2 weeks, we have those talks to look forward to. Thanks so much to our speakers and to those at RTI behind the scenes that are making this happen for us today. And Josh, I'd like to hand it back over to you for any concluding remarks you might have. Thanks so much and have a good day, everyone.
All right. Thank you so much, Tamara. for finishing everything out. Obviously, thank you to Dr. Noel and Dr. Taylor for their presentations as well. We are going to go ahead and wrap things up for the day. In doing so, we do want to make sure everyone knows that we are recording all of these modules. We have completed the first 2 recordings and have them back up on the web page. Julian is going to post in the chat pod momentarily the web page that will take you back to the main workshop series page. There you will be able to access both Module 1 and Module 2. We are working on Module 3. So if you were not able to view those modules, or if you would like a refresher, please go ahead and navigate back to that page once we are done, and you will be able to access, again, Module 1 and Module 2. Hopefully we'll have Module 3 up there by the end of the week, and then with this Module 4, we should have it up by the end of the following week. We are working hard to make sure that they all get back out to you, the public, as quickly as possible, so we definitely appreciate your patience. Also, before you leave, we would like to push you to our customer satisfaction survey. You will find that below the closed captioning pod on the bottom right-hand corner of your screen. So if you could take a few moments before you leave today to go through that quick survey, we would definitely appreciate any feedback that you have, not only about the series as a whole, but also the brand new platform that we are bringing all of our events to you on as well. Just looking through some of my notes. This is, again, Module 4. As Dr. Moretti was explaining, we will take next week off, and then we will have our following webinar on June the 12th. That will be Module 5, then June 19th for Module 6, and June 26th for Module 7. We will be finishing up those registration pages here in the next few days, hopefully, as well. That way we can get those back out to you to allow you to register before the event. Again, thank you everyone for joining us today. These webinars and workshops are simply not possible without your attendance and continued support, so thank you very much for that. Again, thank you, Dr. Moretti, for everything that you've done putting this series together, as well as our presenters today, Dr. Noel and Dr. Taylor, for your presentations. We hope everyone has a great afternoon, and we can't wait to see you for Module 5 back here on June 12th. Thank you very much, and have a great afternoon.
Recording has stopped.